All right, welcome to session 1B. It's nice to see some of you. We will start with the presentation from Dhanushka Devendra. He is from the Institute of Oceanology. Hello, Dhanushka.
Yeah.
Hi.
Can you hear me?
Yes, we can hear you.
Okay. And I will share my screen now.
Yes, great.
Give me 1 second, please. Okay. Yeah. Can you see?
Yes.
Okay.
Yeah, great.
Good morning, all. I'm Dhanushka Devendra from Sri Lanka. Currently, I am reading my PhD at the Institute of Oceanology. Today, I'm going to give my speech under this topic, late Quaternary Paleoceanography of the northeastern Indian Ocean, implication for the primary productivity and deep water changes. For this research work, we used 2 sediment cores from the northeastern Indian Ocean. Some of the data which I am going to present, and during this presentation, I already published in different journals. So this is the outline for my speech. Then I jump into the introduction. So I would like to start this presentation by introducing the Indian Ocean dynamics. Indian monsoon is the most important factor which control the Indian Ocean dynamics. We have 2 types of Indian monsoon, Indian summer monsoon and Indian winter monsoon. So among these 2 monsoon periods, Indian summer monsoon is the most important one because it gives more precipitation to the Indian subcontinent. This precipitation directly affect for the regional environmental changes within the ocean water body because of the more freshwater dilution, and as well as by more sediment transportation through the main rivers which open to this region. As I explained, Indian summer monsoon, we can consider it as a major factor to control the ocean salinity within the region, and it simultaneously affect for the regional environmental changes. It means hydrology and surface water productivity, and then oxygenation as well. Apart from the Indian monsoon, thermohaline circulation also play an important role because North Indian Ocean sector, the bottom water of the Indian Ocean sector originated from the time to time it's changed. North Atlantic, from the near North Atlantic deep water, as well as from here circumpolar deep water, which containing more Antarctic bottom water. So I will explain my results regarding to Antarctic bottom water and North Atlantic bottom water changes with the millennium time scale. So the main reason to conduct this research work, we don't have enough knowledge for this region regarding to paleoceanographic changes, because we have only few researchers have been conducted the kind of research. So we have a very big research gap. So that's the main reason to conduct this research for this particular region. We have lucky enough to answer these 2 questions during this research work: how the surface water mass evolution in the northeastern Indian Ocean, and how the bottom water ventilation and circulation changes, and how it's related with the Antarctic bottom water and North Atlantic deep water. For this research work, we used, as I earlier explained, 2 sediment cores, one from the Central Bay of Bengal, another one from the Andaman Sea. Those belong to the northeastern Indian Ocean. So, but here I'm going to present only data from this sediment core. This is the core description, and we analyze 52 sediment samples for the reconstruct the paleo environmental changes, and these are the proxies we used. And we identified 40 benthic foraminiferal species. I just put some species here to see. Then I move to the main part. Here you can see our age model, age depth relationship. In order to produce this age model, we used 7 age type points. You can see the dating information from this table, and we have the oldest sediment of this core. It's about 56 kilo years, and these numbers indicating the sedimentation rate between the 2 age type points. So you can see here, it has a clear spike. I mean, sedimentation rate is very high during this particular period. It almost belongs to glacial period. The possible reason to have this much of higher sedimentation rate during this period, it is because of the lower sea level. Due to the lower sea level, the distance between our co-location and the river mouth, which open to the Indian Ocean, getting more reduced, and it helps to transport more sediment to the our co-location. So we have this much of peak during this period. Then I move to the explain about the deep and surface water mass evolution in the northeastern Indian Ocean. First of all, I would like to begin with the surface water mass evolution. So in order to provide a better comparison, I used some reference course from the vicinity area, and you can see about this figure. It is about the delta 18 variation within the surface water mass. So all the figures shows almost similar variation trend, but the magnitude is different because of the freshwater dilution. Freshwater is the most important factor for this particular region, and we calculate the delta 18 shift between Holocene and last glacial maximum. So the value exceeding the ice volume effect by nearly 0.6, and it is because of the decreases in glacial sea surface temperature here by nearly 3 centigrade. 3 centigrade. So we calculate this value by using Shackleton thermal equation. Then this figure is about the benthic stabilized changes. So same as for the surface water, we did the calculation of delta 18 shift for the last glacial and Holocene. So that value also exceeding the ice volume effect by nearly 0.5, which corresponding to the nearly 2 centigrade cooler glacial deep water than the Holocene period. So, and this much of cool may be related to the influx of circumpolar deep water with higher proportion of Antarctic bottom water, which containing low delta C13 signals, because Antarctic bottom water containing more or less delta C13 signals. So we can check here. Delta C13 records during the glacial period, we have it's very low value. So it means northeastern Indian Ocean, especially this sector of the Indian Ocean, deep water containing during the glacial period more Antarctic bottom water. Then this figure, it's about the neodymium isotopes. Neodymium isotope, it's a very important proxy to identify the water mass changes, especially North Atlantic deep water changes. So the most negative value indicate the more North Atlantic deep water propagation to the Indian Ocean, and most positive value indicate the low North Atlantic deep water propagation. So during the glacial period, I already explained that northeastern Indian Ocean experienced more Antarctic bottom water current, but here also it shows that during this period it has a more negative value. It means low North Atlantic deep water propagation. But when it comes to the interglacial period, the situation completely changed. You can see the value become more negative. It means more North Atlantic deep water propagation to the northeastern Indian Ocean, and usually North Atlantic deep water containing more delta C13 signals than the Antarctic bottom water. So we can check the delta C13 signals as well. So you can see here, it also show the increasing trend. It means our conclusion, I mean, our identification is correct here. Okay. Apart from that, we calculate the delta C13 difference between planktonic and benthic foraminifera. So this usually we can use this proxy to identify the ocean water circulation. So higher value indicating the low circulation or weak circulation, and more low value indicating the good, I mean, enhanced and active circulation. I want to add another point here. During the higher circulation period within the ocean water body, if it has a higher circulation, it means it should give the more carbon dioxide to the atmosphere because of the oxidation process. So I plot the atmospheric carbon dioxide partial pressure as well from the Antarctic ice core. So during the weak circulation, we have relatively low carbon dioxide partial pressure within, in the atmosphere. But when it comes to the active circulation period during the interglacial period, we can see, of course, we have higher atmospheric carbon dioxide partial pressure. So circulation is very active during this period. And apart from the above stabilized of records, we performed the Q mode factor analysis as well. So we identified 3 factors, and according to these factors, we identified 3 different paleoceanographic stages. It's almost aligned with the marine isotope stage 1, 2, and 3. Stage 1, it's dominated by Polynia species, which indicate the low surface water productivity and active bottom water ventilation. And stage 2 dominated by Vigirina peregrina, which indicated higher surface water productivity and low bottom water ventilation or oxygenation. And during this stage 3, it is dominated by Melanosis species. So we can use this species to indicate the intermediate to high surface water productivity as well as moderate bottom water ventilation. This faunal records is almost tally with our stabilized of records, which I previously explained. And this is my last slide. During the glacial period, our co-location, it means a northeastern Indian Ocean, experienced enhanced surface water productivity, and major portion of the bottom water containing Antarctic bottom water with low delta C13 signals. And our the ocean circulation during the glacial period is very weak, so hence atmospheric carbon dioxide level also very low. But when we come to the interglacial period, the ocean circulation is very active, and then atmospheric carbon dioxide level also very high, but the surface water productivity very low, and major portion of the bottom water containing North Atlantic deep water with more delta C13 signals. And our records indicate that glacial deep and surface water, the temperature of the glacial and glacial deep and surface water 2 to 3 centigrade cooler than the Holocene. Yeah, these are references, and thank you. That's all.
Thank you very much, Dhanushka. Perfect timing. Does anyone have any questions? Okay, so maybe I have a short question to you, Dhanushka. If you could go back to the place where you told about the increase of sedimentation, the rapid increase of sedimentation. I was just wondering, were there any specific species of foraminifera during that time, or were there a difference in the assemblage, or did you find?
Actually, I cannot say like that, because we don't have such a, we did not identify any special occurrence of any foraminifera during this period. But the only sedimentation rate is very high because of this almost belong to Heinrich Stadial 2. I think we don't have special foraminifera, any group increase in trend during this period.
And the abundance, was it smaller because of the increase of sediment?
Yes. Yeah.
Okay, so everything fits. Okay.
Yeah. Okay.
Yeah. Thank you very much.
Okay. Welcome.
Okay. So the next person is Niccolò Lossi from the University of Milano Bicocca. Welcome, Niccolò.
Hello. Hi.
Hi.
Okay. Can you see my screen?
Yes, we can. Yeah.
Okay. Hello, everyone. I'm Niccolò, and I am from University Bicocca, and I am first year PhD student. Now I'm going to talk to you about the connection between aerosols and between aerosols and climate change. And in particular, we measure the heating rate due to light absorbing aerosols between mid-latitudes and the Arctic region. Well, we know that the climate change is a global phenomenon, but in the Arctic region, the rate of warming is twice as fast as the rest of the planet. And this phenomenon led to the expression Arctic amplification. Arctic amplification is due to local effects and feedback due to anthropogenic pollutants emitted or transported in the Arctic region, and to local forcing at lower latitudes, which increase the transport of heat northwards from the oceans and the atmosphere. One of the less clear aspects in the Arctic amplification is the role of aerosols, and most of the studies are modeling studies. And for example, Schindel et al. Suggest us that the increase in black carbon emissions and the reduction of sulfate emissions from the 70s is responsible for the 2/3 of the total temperature increase of 1.5 degrees in the Arctic region. A more recent study by Navarro et al. Suggests us that the reduction of sulfate emissions in Europe since the 80s led to an average increase of temperature per decade of 0.5 degree. We focused on light absorbing aerosols, and the most important light absorbing aerosols are black carbon and brown carbon. In particular, black carbon is the second largest individual warming agent in the atmosphere after CO2. Light absorbing aerosols can act as local forcers with direct, indirect, and semi-direct effects, or indirectly by means of the energy gradient induced by their forcing at lower latitudes. So the key point is to understand the balance and the most important between local effects and the energy transport due to light absorbing aerosols in the Arctic region. Indeed, the purpose of our research is to verify the existence of an increased energy gradient and consequent atmospheric heat transport northwards induced by heat storage at mid-latitudes due to light absorbing aerosol, which can contribute to the Arctic amplification. In order to test our hypothesis, we took part in 2 research cruises, Arctic Expedition, in 2018 and 2019 between Gdansk and Svalbard Island to experimentally define the atmospheric heating rate due to black and brown carbon and its latitudinal gradient. So on board the Oceania research vessel, in collaboration with the IOPAN, we installed an etalometer to infer equivalent black carbon concentration and absorption coefficients at 7 wavelengths from UV to the infrared region, an high volume sampler by Tecora to collect PTS, total suspended particles on filters, an SPM1 pyranometer to measure global and diffuse radiation, and also a spectroradiometer to infer incident and reflected radiation, but divided into wavelengths. To the rock system, we added also a gimbal for the shift movement compensation and a shadow band for measuring also the diffuse radiation. By means of the experimental measurement of black carbon and solar radiation, we can calculate the heating rate in any sky condition at the time resolution of 1 data per minute. We can also make an apportionment of the heating rate based on sources, fossil fuel and biomass burning, and species, black and brown carbon. Here below, we have the equation for the heating rate, and in the equation, we have the absorption coefficient and the 3 components of the radiation, diffuse, direct, and reflected. This slide is about the results for the outward section of RX 2019. Here we have the path of the cruise. And this slide shows us the latitudinal averages of black carbon concentration from Europe, Gdansk, to the Arctic. We can see that there is a big difference between Europe and Arctic. The European background is more or less 1 microgram per meter cube, while the Arctic background is less than 10 nanogram per meter cube. This value, particularly high, is due to the fact that the ship went in the harbor of Tromsø in north of Norway here. Regarding the radiation, we can see that the global radiation decreases northwards, but the diffuse component of the solar radiation increases beyond the polar circle. We obtain more or less the same results for the return section of AREX 2019. The path of the cruise was a little bit different, but we have more or less the same results. The European background is again 1 microgram per meter cube, while the Arctic background is less than 10 nanograms per meter cube. So there is a concentration at mid-latitudes are much higher than in the Arctic. We have some hotspot in the Arctic. For example, at this latitude, we have relatively high black carbon concentration because there is Longyearbyen that there is the biggest human settlement in the Arctic region. But in general, the Arctic background is very low. Again, the diffuse component of the solar radiation increases towards the north. These results are about AREX 2018, but they are pretty similar. Again, we have global radiation higher at mid-latitudes and black carbon concentration higher at mid-latitudes. By means of the absorption coefficients and global radiation, we can calculate the absorptive direct radiative effect, the so-called ADRE, and the consequent heating rate. ADRE is in red and heating rate is in yellow, and we can see the strong correlation between ADRE and heating rate. This figure is about the outward section, and this is about the return section of AREX 2019, and we can see that consequently the heating rate is much higher at mid-latitudes compared to high latitudes. By means of the ADRE, we can calculate also the joule of energy in joule per meter cube added to the system in some key points of the cruise. We consider also Milan because in the same periods we computed the ADRE also in the city of Milan. And we can see that there are 2 orders of magnitude of difference between mid-latitudes and North Pole in the joule of energy added to the system. At mid-latitudes, we have hundreds, and in the Arctic, we have units. So we can see that there is a great energy gradient between mid-latitudes and the Arctic. If we compare these results with the results of AREX 2018, are pretty similar. Again, we have 2 orders of magnitude of difference between mid-latitudes and the North Pole. So we can affirm that there is a great energy gradient and probably a consequent energy transport northwards. We make also the apportionment of the heating rate according to the solar radiation, because the diffuse component of the solar radiation increases beyond the polar circle. Also the heating rate component due to diffuse radiation increases beyond the polar circle. We have 60% and beyond more or less 90%. So we experimentally calculated the heating rate in any sky condition, unlike the models. And this is very important because models generally worked under clear sky condition. We saw the importance of the diffuse radiation for the heating rate at high latitudes, and this means that can be inherent errors in the models. We make also the apportionment for heating rate based on source and species. And we can see that the biomass burning contribution to the heating rate increases toward the north, and consequently also the brown carbon contribution to the heating rate is higher beyond the polar circle, because brown carbon comes mainly from biomass burning. This is also very important because biomass burning, the contribution of biomass burning probably in the future will increase, because we will have more and more fires in the Arctic region. Such happened in Siberia in 2019 due to the rise of atmospheric temperature. So, in conclusion, we saw that there is a progressive decrease in black carbon concentration northwards and away from big cities. There are some hotspots in the Arctic, like long year bin, but in general, we have very low background concentrations. There is an increase in the contribution of biomass burning to black carbon concentration towards high latitudes. There is a reduction of global radiation towards the north and a simultaneous increase of the diffuse component. And there is a remarkable decrease in ADRE and consequent heating rate moving north, according to the latitudinal behavior of black carbon and radiation. We have maximum values of ADRE and heating rate at mid-latitudes and minimum values in the Arctic. There is also an increase in the biomass burning and brown carbon contribution to the heating rate towards the north. So we can affirm that there is a great energy gradient between mid-latitudes and the Arctic due to light absorbing aerosols, and probably a consequent energy and heat transport towards the north. These results suggest us that the energy gradient induced by light absorbing aerosol at mid-latitudes is more responsible for Arctic amplification than local forcings. And these results agree also with the models which identify an atmospheric energy flux between lower latitudes and the Arctic among the causes of Arctic amplification. This research is only a starting point. Indeed, in future, we will have to study deeper the back trajectories, for example, and we will study the heating rate also in 3 dimensions, including also the vertical dimension. The final goal of the research is to make a relative weight assessment of light absorbing aerosol within the overall heat transport to the Arctic. And why it's so important to study the connection between aerosols and climate change, because aerosols are short-lived climate pollutants. So, for example, we stop now to emit black carbon or other light absorbing aerosols, the climate response will be faster than if we stop now to emit, for example, CO2 or other greenhouse gases. Thank you for your attention.
Thank you very much, Niccolò. Are there any questions? Okay, so maybe I will ask one. Niccolò, could you go to the one of the first slides from results? I was just wondering what happened at 60N. What is the magical thing with the northern latitude, you know, 60N? It's maybe not this one. There was another one that it had a very, yeah, here. So I was just wondering, maybe is it only like I just need to look at the map. Oh, okay, because I was wondering if it's also the effect of the ship going more away from the land. You know, I was just wondering if you have any results on sort of a transit from land to the open ocean, if it has a strong effect or if it doesn't.
Yeah, if you sail near the coast, black carbon concentration are higher. Yeah, for sure. But sometimes in 2018, the ship went near the Greenland coast. Okay. And we have light absorbing aerosol, relatively high concentration, because probably we have some signal from dust. And also dust is a light absorbing aerosols. So we have to compare black carbon and brown carbon also with dust in the Arctic region, because glacials are, there is less ice, so there will be more dust in the atmosphere in the future.
Okay, thank you. And I'm wondering if there are other sources of black carbon than forest fires that are natural in the Arctic, or would it be close to zero if there were no fires and no humans?
In general, it's close to zero, but we have some hotspots in some human fjords, for example, in Svalbard Islands, and we have some kind of transport from mid-latitudes. But this transport of particles is not so important compared to the heat transport due to the local emission at mid-latitudes, because there is a very big difference between emission at mid-latitudes and in the Arctic region.
Okay, thank you very much. Great.
Thanks.
Okay, so now we have Karol Mazanowski from University of Gdansk.
Hello.
Hi, hello.
So I can start?
Yes, but we don't see your presentation yet.
Okay. try to share. How about now?
Yes, but it's black now, but yeah, we can see it.
Okay. So hello, my name is Karol Mazanowski. I'm PhD student at the University of Gdansk, and today I would like to introduce to you the topic of gelatinous zooplankton distribution and diversity of the Northeast Greenland coast. I start with short introduction. The average global temperature has been continuously rising over 2 centuries. The measurement of temperature conducted in Greenland since 1970s confirmed this trend. As a result of this, Greenland ice cover melts, and the inflow of glacier waters to coastal water make them fresher and alter biomass and primary production. In the same time, Atlantic waters warming and salting Arctic waters, and as a result of it, Arctic species communities changes towards boreal organisms, and this phenomena was called Atlantification. The Greenland Sea is the area of cooling and freshening Atlantic waters carried by West Spitsbergen current, which turns westward in the northern area of the Greenland Sea in the French Strait, when it becomes outer East Greenland current. And hence, as you can see, it's forming along North-East Greenland shelf frontal system, boundary between colder and less saline Arctic waters carried by the East Greenland current, and warmer and more saline Atlantic waters carried by outer East Greenland current. And the white space here is, of course, the shelf break. Plankton is a good climate changes indicator, thanks to the feature of this organism. For example, they differ in the waters, and the period of life of this organism is relatively short. And a good example of this is gelatinous zooplankton, here understood as cnidarians and ctenophores, which representatives you can see here on these pictures. Changes occurring in gelatinous zooplankton communities reflected changes occurs in physical properties of water column. However, still is little known about distribution of this fragile and gelatinous organism, which is hard to collect it. So our goals were to characterize gelatinous zooplankton communities and their environmental diverse along the northeast Greenland coast. The sampling were collected over the northeast Greenland coast by Danish scientist groups at deck of research vessel Dana in 2017. Originally, samples were collected from 27 stations. For the purpose of this study, we analyzed samples from 10 stations, which forming two transects across shelf of Greenland, as you can see here on the map. Gelatinous zooplankton were collected among all of zooplankton organisms and were identified to the lowest possible taxonomic level and counted. In the term of colonial organisms, as a one colony was treated, each founded locomotory structure because these organisms have only one substructures. And here we have results. As you can see, there was distinguished 5 water masses. Polar surface water worm and polar surface water was waters of Arctic origin and also upper polar deep water. The Arctic Atlantic water was water masses with contribution both of Arctic and Atlantic waters. And return Atlantic water was the most saline and the warmest masses yes, with Atlantic waters. And here, as you can see on the graph, samples were grouped in terms of similarity of gelatinous community between samples, and this grouping reflects water masses, each of distinguished water masses. Here we can see horizontal distribution of gelatinous zooplankton over the shelf in relation to bathymetric zone. As you can see, the most distinguished from others was off-shelf with significant levels, and was also the most numerous in taxa, while in shelf was the most abundant zone in individuals. Also, we can see here that 2 species is dominant species, Aglantha digitale and Physico-chemical borealis. And Aglantha digitale was restricted to the off-shelf and shelf break, and Physico-chemical borealis mostly to the shelf and coastal. Comparing different sites, samplings, north and south, we can see that north is more abundant in terms of taxa and individuals. 4 taxa were common between both sites, and on the north we find 6 taxa specific to this site. However, statistical analysis don't find this differences significant. And also here we can see distribution of gelatinous zooplankton over the each of 5 water masses. As we can see, Aglanta digitale is presented on all of water masses. The most abundant was in upper pole deep water and return Atlantic water, and Quaternary Paleoceanography was abundant, the most abundant in polar surface water and Arctic Atlantic water. And Echinopsis laurenti was the only species presented at on polar surface water 1. And now I move to conclusion. So water masses and bathymetry shape gelatinous zooplankton distribution as statistical analysis show. And the most frequently recorded species were Protocnida borealis, which is Arctic species, and boreal species Aglanta digitale. In the case of the shelf and the offshore, most taxa reflected the hydrological regime. And as you can see previously, only Aglanta digitale found in return Atlantic water. Abundance was higher above the shelf, and as I mentioned, gelatinous zooplankton diversity was similar among some clinic sites. And that's all will be all. And thank you for your attention.
Thank you very much. Are there any questions to Karol? Okay, so I was wondering if you could go to the conclusions, just one slide before. Yeah, here. Do you maybe have any suspicions why the abundance is higher above the shelf? Is it like normal? Because I'm not a plankton specialist, I'm benthic, so I don't know if it's common.
In this case, it was mostly because of abundance of Protocnida borealis. And here we can find over the shelf the Arctic water masses of Arctic origin. And Protocnida borealis is, as I mentioned, an Arctic species. So probably these properties of this environment help this species to develop. And
so it goes all to the masses, yeah.
Can you repeat?
Yeah, it goes all back to the water masses.
Yes, and also some research found that Greenland, northeast Greenland shelf is in some way connected to Barents Sea and Iceland, and there also Protocnida borealis is recorded in large abundance, larger than in other research sites, regions.
Okay, thank you very much, Karol.
Thank you.
Okay, thank you. And now the last presentation by Marlena Szeligowska. She's from the Institute of Oceanology. Hello, Marlena, you can start. I cannot hear you. Maybe it's the headphones because you are not muted.
Marlena, if you go down to the left corner, there is an options. When you just scroll it, you can select a microphone.
Is it okay now?
Yes.
Yes.
Okay, perfect. Okay, so without headphones anyway. Okay.
Okay, we can see everything.
Perfect. So, hello everyone, my name is Marlena and I'm from Institute of Oceanology, Polish Academy of Sciences. And today I would like to present you my PhD journey, and which is supposed to end with model of plankton and particles in the Arctic fjords. And I could say that I'm halfway there, as my second year, the end of my second year is approaching, but I really still ask myself this question: how can I do that? How can I model composition of plankton in the West Spitsbergen coastal waters? And obviously in this presentation, I will try to answer this question, but I would like to start from the very beginning. So the idea for my PhD project came up during previous research expeditions to the Arctic. So my supervisors came up with this idea because of what they could see in the field. And for those who are not lucky enough to go to the Arctic, we can still observe what's going on there in satellite images, such as this one. So there is a lot of particulate matter coming from land together with meltwaters from glaciers and glacier-fed rivers. And we can observe so-called brown zones in the coastal Arctic waters, such as like in this satellite image. And definitely this has to have an impact on plankton and many organisms in the coastal waters. So there is this intensified runoff because of climate warming, and the concentrations of suspended particles are higher and higher. So this causes so-called water darkening. So the light conditions are not preferable for primary producers, and it has many consequences when we go up the food web. So we studied one fjord in particular. This is Isfjorden. We extensively geared up with many devices, and we could observe this horizontal pattern that close to the glacier or river discharge, the very muddy waters. And there are also open waters where the living conditions for plankton are very different. So muddy waters are marked with brown color and clear waters with blue one in this figure. And there is an example of records from the Arctic, from the glacier influenced waters, and also for open waters. And we can immediately see that they are completely different. But when we go a little bit deeper, here and here as well, we can see more and more differences when it comes to the organisms that we can observe there, and also the color of the seawater. But when we go even deeper, we can clearly notice the process that seems like underwater snowing. And these are actually huge aggregates of dead bodies, of zooplankton, some fecal pellets, also phytoplankton flocks, and also sediment from land. And they are combined together. They form these aggregates that we call marine snow. So here on the right-hand side, I presented like some kind of a schematic representation of what's going on the underwater. But obviously to do science, we need numbers. And here is turbidity, which is one of the parameters that we measured. And to put it simply, it says how dirty is the water. It's like very simple definition, let's say. So I did zoom in on the regions that we studied, and here the three of them here are glacier or river influenced, And here on the right-hand side, there are two regions that are clear, let's say. So definitely turbidity was much lower there. And also we could observe different colors of surface seawater, which is presented on the bottom. And the other parameter that we could measure was abundance of marine snow, so these macroscopic aggregates. And we used underwater camera for these, but it's like very special underwater camera. It's called underwater vision profiler, and it actually saves pictures of different objects that it detects in the water. And here in the middle, we can see like the composition of different pictures of the separate objects that they were present in the water. And in the glacier influenced or river influenced waters, we could see more mineral particles. So the pink one and light green. But when we went to open waters, the clear waters, like there were more dead bodies or phytoplankton flocks, so more organic matter. It was much more important than inorganic matter. And when it comes to the abundance of marine snow and zooplankton in the glacier influenced waters, especially in the Biellefjorden, in the Glacier Bay, there was high abundance of marine snow, and the zooplankton was not really present in many stations there. But when we go to the open waters, there was much more zooplankton. And we could also see that in the common digital camera that I presented in the beginning. So these are just few results from my last paper published in Science of the Total Environment. But what we were really interested in is how to connect what we can see in the field with how we can model it, how we can represent all of the important processes in the numerical framework, in the equations. So what we did, we divided protists and zooplankton in the taxonomical groups that are used in common biogeochemical models. And this is like very coarse classification. There are only 4 groups of plankton represented in biogeochemical models typically. So there are diatoms, flagellates, microzooplankton, and mesozooplankton. And according to this division, we could also detect differences between muddy waters, glacier-influenced waters, and clear waters. So this says that we could actually use these models, these frameworks that are already there, to represent plankton composition and represent differences between muddy and clear waters in the numerical framework. So there are some groups that are more responsive, let's say more responsive, so they are less abundant in the muddy waters than in clear waters. And we would really focus on these groups. Also, I added gelatinous zooplankton, about which Karol talked about. So what we observed was that gelatinous zooplankton is like very good indicator of living conditions for plankton in coastal waters in the Arctic. And going further, it was just one point of view, the horizontal patterns, but now we want to go deeper in this description, and we want to reveal some vertical patterns in these coastal waters, because we really need to know all of the processes well to do the predictive models, to be able to represent everything in the numerical framework. So we have more fjords. This is Hornsund and area in front of Torelbreen Glacier. And here we also study the same parameters, but we really focus on the vertical distribution of turbidity. So where are the peaks of turbidity? If it's only at the surface, or there are also subsurface turbidity peaks. And we also try to link this with fluorescence of chlorophyll and see what is the difference between peaks depth. Are they together or are they separated? But also we want to follow the what we call depth of turbidity change. So to which point, to which depth, we could say that the water is dirty, that the water is muddy. And if this change in the water conditions of light distribution does this change affect distribution of zooplankton? So this is something that we currently work on, but at the same time we do this modeling. So I applied for short-term research grant, and I went to Germany from November till February for 3 months. And I worked with Dr. Deborah Bencourt from Helmholtz-Zentrum Heron. She's from a modeling group that works on the changes in the Arctic Ocean. And I used a hydrological model coupled with biogeochemical model. So the framework that she works with, and she really nicely introduced me to this whole modeling. And this is like the schematic representation of the model that I work with. So all of the boxes represent some component of the seawater. So there are like there is phytoplankton, zooplankton, but also nutrients, detritus and sediment, macrobenthos. And what I added within the scope of my 3-month stay in Germany was SPM, so suspended particulate matter. It is especially important for glacier influenced waters. So this is really the point of my stay there, to represent this suspended particulate matter in the model and interactions with different boxes, let's say, in the model. And I also introduced meroplankton because in the coastal waters, it is really important constituent of plankton community. So I found it really important to link this benthic and pelagic environment. So to sum up, I studied horizontal and vertical environmental gradients, and at the same time I prepare numerical framework in 1D. It's still work in progress, but I also have future plans, and this is application of this new polar version of eCosmo that I work with in 3D modeling of Svalbard coastal waters. So I'm going to go back to Germany for much longer to do this 3D modeling. And the more I go into this modeling, I think that it takes a crazy person to do that, but here I am. So just, yeah, wish me luck with that. Okay, so thank you very much. This is the funding for my projects, and yeah, I'm open for questions.
Thank you very much, Marlena. Good luck.
Thank you.
Are there any questions? Okay, so I have, I think, a short one, but I don't know. So you don't have any results regarding the turbidity depth yet, yes? You will just work on it. You don't have it yet.
I mean, when it comes to the turbidity, we have some patterns when it comes to the turbidity and other environmental conditions like salinity, temperature, fluorescence of chlorophyll, and also some optical properties, the concentration of particles from 2 laser counters, but we were not able to somehow like find the correlation between these patterns and zooplankton distribution. So this is something I work on currently. So yeah, this is work in progress, but also we could divide regions according to the environmental conditions. So for example, here we have first clear waters, Adventfjorden and Isfjorden proper, where the turbidity is like really low and fluorescence of chlorophyll is high. So the peaks are very separated. But for example, when we go to the area in front of Torrelbrunn Glacier, and in Horsund, the peaks are at the same depth. So there are some subsurface plums that introduce this suspended particulate matter to the coastal waters, and we could see that here there is this subsurface plum. We could also see this in the temperature profile and seawater stability. There is this parameter and Brand-Weissala frequency. So we use that to see where the plum is introduced, so where the fresh water is introduced from the glacier. Yeah, so this is work in progress, yes.
Okay, thank you very much, Marlena. And this is all we have for this session 1B. And now we have a 10-minute break. Thank you very much all for coming and for giving your presentation. It was very nice. See you soon.
Hello, it's 11:10. I would like to welcome you on session number 2. And the first presentation will be presented by Mattia Fragola from University of Salento. Are you with us? Unmute yourself, please. Hello.
Hi.
Oh, now we
Good
can hear you.
Okay.
Hello. Is your presentation ready?
Yes, I will try to share.
Mm-hmm.
Don't know if you can see it.
Yeah, it's loading.
Okay.
Okay, now we can see.
Okay,
So
perfect.
you can start. The floor is yours.
Okay. So my name is Mattia Fragola, and I'm a PhD student in physics and nanoscience from University of Salento in Lecce, Italy. And I'm going to talk about seasonal variability of the airborne eukaryotic community structure at a coastal site of the central Mediterranean. So aerosol, also known as particulate matter or PM, includes all the solid and liquid particles of microscopic size which are suspended in the atmosphere. Speaking of PM10 in particular, we can say that 10% of the total aerosol mass is made up of biological aerosol or bioaerosol, which is all the airborne particles coming from biological organisms, including living and dead microorganisms, such as we can see in this figure on the left, mostly bacteria, eukaryotes like plants and fungi, but also viruses and archaeobacteria. So in this study, we used the high-throughput sequencing of 18S ribosomal RNA gene to draw up a preliminary local dataset on the seasonal dependence of the airborne eukaryotic community biodiversity detected in 37 PM10 samples that we collected from July 2018 to June 2019 at a coastal site in southeastern Italy. So we decided to focus on the characterization of the structure and seasonality of airborne viridiplankton fungi at the genus level, since they were the main components of eukaryotes in our samples. We also investigated the potential impact of long-range transported airflows and local meteorological data on the eukaryotic community structure. Moreover, we also used some statistical techniques to study the relationships among plant and fungal genera, and with meteorological parameters and PM10 mass concentrations. Finally, we also gave special attention to potential pathogenic fungi and plant-derived allergens due to their potential harmful effects on both plants and humans. So all the PM10 samplings were performed on the roof of Mathematics and Physics department at University of Salento as part of aerosol and climate laboratory activities and within the ACTRIS infrastructure. This is a monitoring site that can be representative of the central Mediterranean basin because of its geographical location. In fact, it is significantly affected by long-range transported air masses from surrounded countries and the Mediterranean Sea itself, alongside pollution and bioaerosol sources as shown in previous studies. So we used a dual sampler shown in this figure to collect by gravimetric method 37 PM10 samples on Teflon filters from July 2018 to June 2019. And then these environmental samples were processed through metagenomics analysis, which means that DNA was extracted from each sample, and then a target gene, in this case 18S ribosomal RNA gene, was then amplified, and the obtained amplicons were sequenced on a high-throughput machine, and finally the sequenced reads were processed through a robust bioinformatics pipeline for the taxonomic characterization and classification of eukaryotes in our samples. So the metabarcoding approach allowed identifying in our PM10 samples 4 eukaryotic kingdoms, which were Viridiplantae, fungi, protista, and metazoa. This figure shows us their main percentage contribution on a logarithmic scale across the 4 seasons, in addition to the corresponding contribution due to the unclassified eukaryotic kingdoms. We found 2 prevailing eukaryotic kingdoms in our samples, Viridiplantae, or green plants, and secondly, fungi. So we decided to focus on these 2 kingdoms for further taxonomic characterization. At the phylum level, we can see that within Viridiplantae kingdom, we found 2 phyla, Streptophyta and Chlorophyta, with the former being the prevailing one across the 4 seasons. While speaking of fungi, we identified 3 phyla, Ascomycota, Basidiomycota, and Microsporidia, with the former 2 being the predominant ones, and with the Ascomycota in particular being the most abundant across all the seasons and mainly in summer. So we moved on to analyzing the biodiversity seasonal dependence of the 46 Streptophyta and 22 fungi genera in our samples. As we can see from this figure on the left, the Shannon index representing species richness and Simpson index representing species evenness for both plant and fungal genera were strongly affected by seasons. We then focused on the study of the 12 most abundant and pervasive Streptophyta and Ascomycota, Basidiomycota genera across the seasons. And we can see from this figure on the right that both fungal and plant genera varied significantly along with the seasons themselves. In particular, we found that for plants, Brassica and Panicum were the most abundant genera in winter and summer respectively, while Olea was the most abundant genus in spring and autumn. Speaking of fungi instead, Botrytis and Colletotricum were the predominant Ascomycota genera, whereas Cryptococcus and Ustilago were the prevailing Basidiomycota genera. So from these 2 other figures, we can also see that fungal and plant genera not only varied along with the seasons, but also from one sample to another, day by day. And looking at those samples with an atypical taxonomic structure with a prevailing single genus, we were also able to study the impact of long-range transported airflows on the sample taxonomic structure. For example, speaking of Viridiplantae, we found an atypically high brassica relative abundance in winter samples 7 and 8, which was probably due to the advection of air masses from Eastern Europe, from which this genus is native. And speaking of fungi, we found an atypically high Justilago abundance in spring sample 11, probably due to the advection of air masses from Northern Africa and the presence of desert dust on the sampling day. So, as already mentioned, we used statistical techniques like Spearman correlation coefficients to highlight relationships between the 12 most abundant and pervasive plant and fungal genera, and with meteorological data and PM10 mass concentrations. Here we can see some of the most significant Spearman correlations for plant and fungal genera, in particular for the 3 most abundant plant and fungal genera across seasons. So we found that Brassica genes for plants was correlated significantly with atmospheric pressure, while Olea was correlated with relative humidity and cumulative rain, and finally Panicum with temperature. Speaking of fungi instead, Botrytis was correlated with wind speed, while Fialavia was correlated with PM10 mass concentration. Finally, we also detected some potential pathogenic fungi genera in our 37 samples. In particular, we found mostly phytopathogenic fungi genera that can negatively affect the production of various crops. As we can see from these 2 figures, that represent the damaging effects of Botrytis fungus on strawberry flowers and fruits, and the damaging effects of Fusarium and Colletotrichum fungi on avocado fruits. Plus, we also identified 10 out of 12 most abundant and pervasive Streptophyta genera that can be classified as allergen-producing plants because of their pollen that can cause respiratory diseases. And these indicate a massive and constant presence of aeroallergens in the study area. So to conclude, we found that Viridiplantae and fungi were the most abundant eukaryotic kingdoms in our PM10 samples. And Streptophyta was the predominant plant phylum, while Ascomycota and Basidiomycota were the predominant fungi phyla. Plant and fungal genus community structure varied not only day by day, but also with seasons in all our samples. And analyzing samples with an atypical community structure, we were able to infer the potential impact of long-range transported airflows on the sample taxonomic composition itself. Few strong positive relationships among genera and meteorological data were found, in contrast to the strong seasonal dependence of the genus relative abundances. Finally, as already mentioned, 6 potential pathogenic fungi genera were detected in our samples, while 10 out of the 12 most abundant and pervasive plant genera we identified were classified as a potential source of aeroallergens at the monitoring area. So thanks for your attention.
Thank you, Mattia. And now it's
Thank
time
you.
for questions. I see Marta has one.
Yes, hi. I really liked your presentation. I
you.
saw that you focused on the eukaryotic organisms, but have you considered including cyanobacteria to your analysis?
Yeah, there were previous studies about the prokaryotic community at the same study area. And we found also that the most abundant phyla and species of bacteria varied along with the season and were also affected by the long-range transported air masses from surrounded countries, as for the eukaryotic communities.
Thank you. Thank you so much.
You're welcome.
Are there any more questions? Okay, thank you, Matija. And another presentation will be presented by Mateusz Pikula from University of Gdansk. Are you with us, Mateusz?
Yes, I am. Hello. I hope I can hear each other.
Yeah, try to share your screen.
Yeah, I'll try to share the screen. I hope you can see it.
Yes, now we can see everything is good, so the floor is yours.
Okay, so hello everyone, my name is Mateusz Pikula, and today the title of my presentation is Molecular Markers in Evolution and Taxonomy of Gyrodactylus gasterostei, Glacier 1974. Presentation schedule is as follows. First, I would like to start with introduction and aims of my research projects, moving on to materials and methods, results, discussion, and ending my presentation with conclusions and bibliography. So a short introduction, what is Gyrodactylus? Gyrodactylus is a genus of small parasitic flatworms which infect a variety of fishes from multiple fishes family. The first species of Gyrodactylus was described nearly 200 years ago by Nordman, and it was called Gyrodactylus elegans. The deteriorating influence of Gyrodactylus on fishes can be exemplified, for example, using the Gyrodactylus salaris example, which was responsible for a series of epidemics which struck salmon farms located in Norway in 1970s and 80s, which were later termed gyrodactylosis. So in short, they are not only the scientific curiosity, but also a pesky troublemakers. As I said before, they were described nearly 200 years ago, and for the most part, their identification was based on the morphological differences in both shapes and sizes of marginal hooks and hamuli. However, the last 25 years saw the introduction of molecular analysis, genetic markers, and genetic research into identifying the Gyrodactylus species. And with over 480 species described out of 25,000 assumed, this genus, Gyrodactylus, seems to be in dire need for a systematic restructurization and clarification using aforementioned genetic markers and new technology that are available for us today. And one of the species that lacks this genetic description, this detailed genetic description, is Gyrodactylus gasterostei, which was described in 1974 by Glacier in Tribunary of Elk in Eastern Germany. It exhibits morphological resemblance to other closely related Gyrodactylus species, such as Gyrodactylus afea or pungiti, and has already been reported in several countries across Europe, for example, in Poland. And it's believed that its specific host is three-spined stickleback, which is Gasterosteus aculevanthus. So moving on to the aims of my research project, as I mentioned before, because Gyrodactylus gasterostei lacks this detailed genetic description and investigation, my research project's main aim was aforementioned investigation of evolution and phylogenesis of Gyrodactylus gasterostei using two genetic markers, ITS-rDNA and COX-1 gene. And said goal included, for example, estimating an approximate age of Gyrodactylus gasterostei, describing the possible origin place for this species, and determining its phylogenetic positions among other Gyrodactylus species. So materials and methods. First, in order to begin the project, I needed to collect my samples, and the samples were collected from the fishes, which were obtained from my department collection, which were collected from 16 locations, mostly located in Pomorskie Voivodeship, with 5 locations lying outside of Pomorskie Voivodeship, as you can see on this slide. Here is better visualization of location of each sampling locations. As you can see, 5 locations were located outside of this cluster of locations located in Pomorskie Voivodeship, which some of them were outside of Poland, while others were located inside Poland. Moving on to genetic markers, as I said before, I used 2 of them. They were my main tool for this whole research project. One was a nuclear marker, which was ITS-rDNA, and the second one, mitochondrial marker, was COX-1 gene. They served as both markers for with COX-1 serving as an intraspecific level marker, while ITS-rDNA serving as interspecific level marker. Research procedures basically could be divided in two parts. The first part was laboratory research, which in turn was divided into six steps. The first step was specimen collection, moving on to the digestion, DNA amplification, gel electrophoresis, DNA purification, and ending with DNA sequencing, which was executed by the outside company. While the second part, the bioinformatic analysis, was conducted using numerous programs, but the four main programs I used were MEGA7, POPART, Vienna RNA Web Service, and BEAST program package. So moving to results, the species identification was the first step, results. As you can see, over 200 specimens of Gyrodactylus were collected. Not all of them were analyzed. In total, we obtained 34 sequences of ITS-rDNA sequences and 85 sequences of COX-1 gene region. During the course of the project, we identified 8 different species of Gyrodactylus, with 3 of them showing significant deviation from the known ITS-RNA sequences known for this species. These species are marked in red in the table on your right. For Gyrodactylus gastrostei, this identification was completely successful. The obtained sequences of ITS-rDNA were identical to the ones we found, for example, in the gene bank. And the species Gyrodactylus cf was used as an outgroup for Gyrodactylus gastrostei. Moving on to haplotype diversity, haplotype can be loosely defined as a variant of gene sequence with single nucleotide polymorphism differentiating one variant from another. In case of COX-1 gene, we've obtained 7 haplotypes, with 5 of them being completely new haplotypes. And as you can see, there are some differences between each haplotype, some small differences, but noticeable. In total, we obtained 71 sequences of COX-1 gene, and they represented the following haplotypes, with haplotype 2 and 3 being the most numerous one. Haplotype 2 represented by 44 sequences, while haplotype 3 represented by 15 sequences. One sequence was found to be heterogeneous, which was a mixture of haplotype N8 and 10. And as you can see, as I mentioned before, haplotypes 2 and 3 were the most abundant one. And because of their abundance, they formed so-called haplogroups, and they were located in center of each haplogroup. The haplogroup 2, which consisted of haplotypes 6, 9, 10, 1, and 2 located in center of it, and haplogroup 3, which consisted of haplotypes 7, 5, 4, and 8, and 3 located in the center of it. Some haplotypes are yet to be found. For example, the haplotype termed NZHP2, which connects the haplotype network for Gyrodactylus gastrostei to its closest outside species, Gyrodactylus cf by approximately 166 nucleotide substitutions, as you can see on this figure below. Here's the map of the haplotype distribution across the Pomorskie Voivodeship and several other locations. As you can see, the haplotypes for haplogroup 3 were characteristic for Vistula tribunaris, while the haplotypes for haplogroup 2 were characteristics for the tribunaris and rivers for Pomorskie Voivodeship in general. As I mentioned before, I also I used the program to generate tRNA TRP structure, or tRNA for tryptophan structure, for 4 different species. For Gyrodactylus gastrostei, there were 2 variants termed HP2C5 and HP2T5, which differ by single point mutation, marked here in this figure by a red frame. I also conducted the molecular clock analysis, which indicated that the most recent common ancestor of all, quote, living COX-1 gene haplotypes for Gyrodactylus gastrostei emerged approximately 60,000 years ago. But the split between 2 close related species, Gyrodactylus gastrostei and Gyrodactylus cf, may have already begun around nearly 3 million years ago. Here's the whole picture of a phylogenetic tree generated using aforementioned molecular clock analysis method. As you can see, the Gyrodactylus gastrostei is indeed a young species compared to other more older species such as Gyrodactylus Lossi or Gyrodactylus salaris, with its age estimated, as I mentioned before, something between 3 million and 60,000 years ago. So moving on to the discussion, As I mentioned before, we obtained seven haplotypes, with 5 of them being completely new, which brings the total number of known COX-1 gene haplotypes for Gelatinous gastrostei to 10. This is a relatively low value, which may confirm the assumption that were proposed for this species over 20 years ago, that this species favor asexual reproduction over sexual reproduction. Also, there were no more than 2 haplotypes in every location, and except for Cardiff, these haplotypes followed a strict pattern, with one being local, which means that it was found only in this certain location, while the other was a dominant one. In comparison, for another Gelatinous species, Gelatinous arquatus, the number of COX-1 gene haplotypes found in one location could be as high as 40, and the total number of haplotypes known for this species is 131, which is 13 times more than for Gelatinous gastrostei. As I mentioned before, molecular clock analysis confirmed our previous assumption that Gelatinous gastrostei is a relatively young species. Our initial proposals were put the age of Gelatinous at 30,000 years ago. However, the final results were 60,000 years ago. Still, it works in favor of our previous statement. Obtained data also confirmed the so-called Doggerland hypothesis, which states that Gelatinous gastrostei may have originated on an ancient landmass called Doggerland, which existed for roughly 10,000 years ago during last glacial period. The map of the Doggerland can be seen at the right part of the screen. So moving on to conclusions, Gelatinous gastrostei shows an excellent example of a young ectoprise-sized species, which most probably underwent post-glacial expansion during last glacial period, originating probably in ancient land of Doggerland. Its low number of COX-1 gene haplotypes found in general between 10 and in any given location with a maximum of 2 may indicate that Gelatinous gastrostei is not only a young species, but it's also a species that favors asexual reproduction over sexual reproduction, which this statement can also be supported by the dominating character of haplotypes 2 and 3, which were represented by approximately 84% of the obtained COX-1 gene sequences for this species. With bibliography for the presentation I just presented. Thank you very much for your patience, and if there are any questions, I'm happy to answer them.
Thank you, Mateusz. Are there any questions to Mateusz? Okay, so I would like to thank you very much.
Thank you very much.
And next presentation is by Aleksandra Stodolna from University of Gdansk. Hello.
Hello.
Okay, we can hear you well.
Okay, can you see my presentation?
Now, yes. Okay, everything is fine, so the floor is yours.
Thank you. Hello, everyone. My name is Aleksandra Stodolna. I'm in my last year of my master's degree, and I would like to share some information linked to my thesis. So the subject of my study is cloning, overproduction, and purification of Escherichia coli RNA polymerase sigma factors. And I would like to start by explaining some of the basic information linked to my work. So a model organism is an organism that is studied to understand some biological mechanisms that can provide an insight into the workings of other organisms. The best and most studied prokaryotic model organism is Escherichia coli that has been studied for over 60 years. E. coli is a common gut bacterium. This bacterial model organism allowed many scientific discoveries such as DNA replication, transcription, and molecular cloning. These processes play an important role in today's biotechnology. With addition of fast growth, low cost, and ease of manipulation, E. coli is a great laboratory staple. As I mentioned, transcription is one of the processes that were discovered thanks to E. coli. This process involves unwinding of the double-stranded DNA and the formation of new RNA strand that is complementary to the DNA strand. Later, the RNA strand is used to translate the information it's carrying into amino acid sequences that form proteins, which are later used to regulate many mechanisms inside every living organism. Transcription requires presence of an enzyme that starts the whole process. This enzyme is called RNA polymerase. It was first found in E. coli, and there is only one RNA polymerase in prokaryotic cells. It is built of 5 subunits. In bacteria, there are 2 alpha subunits, 1 beta subunit, 1 beta prime subunit, and omega subunit. They form a so-called RNA core polymerase, but only when a sixth subunit called sigma is bound to the RNA core polymerase, the transcription process can start. Sigma subunits, or sigma factors, that are the main subject of my research, are proteins that enable binding of RNA polymerase to specific sequences that are called promoters. Promoters are sequences of DNA in the beginning of a gene that can encode a variety of proteins, helping with adapting to the environment. Due to diversity of environments the bacteria can live in, they have developed many variants of sigma factors that bind to specific promoters depending on the current conditions they are in. All bacteria have specific sigma factors. For example, Mycoplasma genitalium that lives in a relatively constant environment has only one sigma factor, and bacteria that live in soil and water, meaning they are exposed to a lot of environmental changes, can have up to 60 sigma factors, like Streptomyces colicolor. Escherichia coli that lives in large intestine has 7 sigma factors. There is a sigma factor called sigma D that is active when bacteria are in normal growth conditions without any stress factors. One is called sigma S that helps activate gene transcription when bacteria are in stationary phase, meaning that the growth rate and the death rate of bacteria are equal. Another one, sigma E, is active when bacteria experience some stress factors like oxidative stress or extreme heat shock. There's also one called sigma F that is responsible for synthesis of bacterial locomotion organelle. There are also sigmas that are responsible for metabolism or assimilation of specific nutrients like iron by sigma Fec-I or nitrogen by sigma N. And last, there is one called sigma H that is active during exposure to heat. Molecular cloning is a process that involves joining of 2 DNA fragments that can be later inserted into bacteria. To conduct this process, we need a fragment of DNA that we want to insert into bacteria and a vector that can carry this sequence. Vector or plasmid is a usually circular DNA molecule that has the ability to carry genes and replicate them in bacteria. Additionally, it has an antibiotic-resistant gene that will come in handy during the last step of molecular cloning. Next step involves cutting the DNA sequence and the plasmid with specific enzymes in the same way so they are compatible with each other. After that, we can join the 2 fragments in a process called ligation. The plasmid now carries the DNA sequence of our interest, can be inserted into bacteria during a process that is called transformation, during which bacteria uptake DNA from the environment. Last step of molecular cloning is selection of bacteria that carry the plasmid with inserted DNA fragment. It is relatively easy because the plasmid we used had an antibiotic-resistant gene. That way we know that the bacteria that grow on a medium with specific antibiotic can only be the ones that have the plasmid. After successful cloning, we can use the transformed bacteria that carry DNA of our interest to overproduce the protein that the DNA encodes. Overproduction of proteins involves using an expression vector that allows this process. These kind of vectors have special promoters that allow production of the target protein in large amounts. The first step of protein expression is transforming the bacteria that specialize in protein production with the expression plasmid that carries the gene of our interest. Next, the bacteria start to produce the protein in large amounts. Next, we need to purify the obtained protein from bacterial cells and other impurities. To obtain purified proteins, we need to disrupt the bacterial cells during lysis so the protein can get out of the cell into the solution. To separate the protein from bacterial cells, we spin the sample at high speed. This is called centrifugation. Our protein is found in solution and the remains of bacterial cells are at the bottom because they are not soluble. Now we start proper purification by loading our sample onto a special column with a filter and the resin at the bottom. The filter and the resin act as a barrier. Proteins are usually tagged, meaning they have special sequence bound to them, which helps with protein cleaning. This tag binds our protein to the resin. During washing, other proteins and impurities can flow through the filter and resin, leaving only our still bound protein. Lastly, we need to remove our protein from the resin. For this purpose, we use special substances called buffers that have the ability to detach our protein from the resin and elute it into the solution. After completing these steps, we can concentrate our proteins to obtain higher yield in lower volume. This can be achieved during centrifugation in special tubes with membranes that our protein won't be able to pass through. Lastly, we need to evaluate the concentration of our protein, which is crucial for the later use of purified proteins. Each step of protein purification needs to be monitored to check if the protein isn't lost during the process and to check for any impurities. This is usually carried out with SDS page. This is the method that allows visualization of our samples on special gel. The gel is placed in a buffer and the samples of proteins can be loaded onto it. It is important to also load a marker that we can compare our samples to because it shows bands at specific sizes. Then we can run our gel with applied electric field. Proteins during this reaction have negative charges, so they migrate towards the positive electrode. Last step of SDS page is staining the gel, which allows visualization of the separated proteins. After that, we can see proteins that appear as bands within the gel. Here I would like to present the results of my work. This is a stained gel after SDS page. First, we have a marker that helps us determine the size of proteins. Then we have our target protein that has the tag that helped during purification. And the rest of the bands are impurities. So we want to get rid of all the impurities and only have our protein in the solution. In this lane is a sample from bacterial culture before the process of protein production has started. Here is a sample from culture after 3 hours after the production of protein has started. This is a sample taken after we disrupted the bacterial cells. Next sample was obtained after we separated the proteins from bacterial cells. So you can see that there are a lot of other proteins in the solution that we need to get rid of. Here is a sample from that was taken after we loaded our protein onto the special column with resin. We can see that unfortunately sometimes some protein is lost at this step. And here is a sample from washing the column with special buffer. We can see that our protein is not found here because our protein has the tag that binds our protein to the resin. So all the other impurities are being washed away except for our protein. Last 3 lanes are from samples after we eluted the protein, meaning the tag was detached from the resin, enabling for our protein to flow through into the solution. At this point, there are not so many other impurities. To get rid of some remaining impurities and the tag that is still bound to our protein, we need to run other purification processes. On this gel, we can see the same marker as previously, our tagged protein, protein without the tag, and the tag itself. First step of this purification process was to run dialysis with our protein. This enables buffer exchange and getting rid of some other impurities. After that, we can finally cleave the tag from our protein. The tag is detached using special enzyme that cleaves the protein from the tag, leaving the protein without any unwanted residues. Last step is concentrating of the protein. So here is a sample before concentration. And here is protein after concentration. So we can clearly see that the yield of the protein is noticeably higher. Here we have a gel that shows the results of my work, purified sigma proteins. So here we have sigma D protein, sigma N protein, sigma S, sigma F, and lastly sigma E. Here we have 5 of the 7 sigmas because I wasn't able to purify all of them just yet because there were some challenging proteins. Sigma H was one of the 2 challenging proteins. During the whole purification process, there were a lot of other impurities that I wasn't able to get rid of using different buffer compositions. Same goes to sigma Faci. There were too many impurities for the proteins to be used in later research. So we still look for other methods to help us purify these 2 proteins. So in conclusion, the protocol that we propose is a new and innovative way of sigma protein purification. There is no protocol that allows purification of sigmas with same methods and buffers for almost all of the sigmas. Our protocol also doesn't include obtaining protein from inclusion bodies, which can be harmful for the protein because they contain denaturated proteins. That is why we chose to work with native conditions. Also, we were able to clone all Escherichia coli sigma factors into an expression vector with his sumo tag that helps with protein solubility and is easily cleaved from the protein. We also successfully purified 5 of the 7 sigma factor proteins, and we are still determined to purify the remaining 2. One of the purified proteins, namely sigma E, has been already successfully tested to check its activity with RNA polymerase. And we still need to verify other purified proteins activity that can show us if the sigmas can bind to RNA core polymerase. Thank you very much for your attention.
Thank you, Alexandra. Are there any questions to Alexandra? Okay, so thank you for your presentation.
Thank you very much.
And I would like to now introduce Wojciech Wesołowski from University of Gdansk.
Hello, everyone.
Hello.
Give me a second, I need to do something. Okay. This. Okay, is it visible now?
Yes, so we can see everything. So the floor is yours.
Okay. Okay, so thank you for introducing me. I'm very glad. My topic is the role of sRNA molecules in an antirepressor-mediated initiation of the phagetic development. First of all, I want to introduce enterohemorrhagic Escherichia coli, which are virulent bacteria. Main virulent factor is Shiga toxin, which was contributed by Shiga toxin converting bacteriophages in horizontal gene transfer way. Infection dose of these bacteria is only 100 bacteria cells, and it's a small number, to be honest, because, for example, infection dose of bacteria from Salmonella genus is 1,000 bacteria cells, so it's 10 times bigger. So it makes enteromorphic Escherichia coli diseases, infections really easy to get. But we don't need to be afraid, to be honest, because in Poland we have only 500 infections per year. And compared, for example, to United States, are 265,000 infections per year. So this differ much in many countries. This is problem of United States, Great Britain, and Germany. And cause diseases are hemorrhagic colitis, hemolytic uremic syndrome, which death ratio fluctuating between 3 and 10%, and thrombotic thrombocytopenic purpura with 30 percent death ratio. So it's quite high. And why are they so high? Because there's no real treatment, to be honest. Antibiotics do not help and mostly makes everything worse. Why is that? To become aware of the problem, we need to understand development cycles of phages. Shiga toxin converting bacteriophages belong to lambdoid phages family, which means that it has two development cycles, lytic and lysogenic. What's important, the main thing here is that the release of Shiga toxin takes place only as a result of bacteriophagetic development, right? In the lysogenic pathway, most of the phage genes is suppressed, and one of the factors that makes prophage induction are antibiotics. So when you give an antibiotics to cure enteromorphic Escherichia coli, it makes everything worse because we have more Shiga toxin with lytic cycle with cell lysis. How it look like in bacteriophages? Bacteriophage feed Phi24b genome. And when, as you can see, we have two operators region. Give me a second when I have, okay. We have two operator regions, and we have this small protein marked by these two orange balls, and it's a C1 repressor, and it represses lytic cycle. It's suppressing the lytic cycle, and and the transcription of genes doesn't occur. Only when in some way gets inactivated, this C1 repressor, everything gets started. Also with Shiga toxin, which is marked here. How does it look like the C1 repressor inactivation? It could take it could be two-way dependent by SOS system it looks like that in some ways host bacteria DNA get damaged, and SOS response is started, and REC-A protein is activated. So this protein can cut inactive C1 repressor. It's C1 repressor is autoproteinized. So he's inactive, and this is quite easy to understand way, because when host DNA got damaged, it's really threat. It's a really big threat that the bacteria will die, and for phage is really the best way is to start lytic cycle and run out from this dying bacteria. But we have other way, antirepressor dependent, and it's this is self in fully in phage way, because antirepressors are phages proteins, and they can bind to C1 protein, stop repression of lytic cycle, so lytic cycle can start. And there's a question that how the antirepressors transcription looks like when they are active, when is any ways to regulate that. So it is we have found two microRNA type molecules. We call them APROI1, which is 29 nucleotides long, and APROI2, which is 39 nucleotides long. They originate from 524B phage and are encoded by sequences located on the opposite strand to AINT and RY genes, which encode antirepressor proteins, right? And so there's from antibiotics, from bioinformatics, in silico, okay, I guess that in silico analysis, they quite high possibility that they can have influence on these genes. But not only that, bioinformatic analysis indicated many other binding sites for APROI1 and APROI2 in the genomes of both bacteriophage and the Escherichia coli host. They occur within bacterial genes coding for important proteins, for example, inner membrane protein and flagellal hook filamental junction protein. There are many of that, but we have chosen them in APROI1. For APROI2, we have chosen ABC transporter TPase, L-fucose isomerase, and palmitoyl acyl carrier protein dependent transferase. Why? Because we highlight these genes because probability of binding hexamate microRNA to these genes is considerably high. But still, it needs a further confirmation, of course, by in vitro research. These proteins take part in metabolism and cell membrane functioning, and disturbing this proteins production could affect on the host life. Go back to the bacteriophage genes. The predicted binding sites of phage origin are localized mainly upstream or within genes encoding antirepressors. When we can see that I have written that they are trans-acting and cis-acting, it means that literally what I have showed here, cis-acting microRNA can affect the the mRNA, which is on the opposite strand, right? And this acting, trans-acting proteins affects any other mRNA in other parts of the genome, and they are not fully complementary compared to cis-acting microRNA, which is fully complementary because of the opposite strand, of course. I think it's clear. And so we have made in vitro research, and it shows that overexpression of these molecules decreases growth rate of host culture after phage infection. Differences are even more visible in higher multiplicity of infection level. For B, it's multiplicity of infection level equals 5. So it means that we have 5 bacteriophages on one host cell. Multiplicity of infection equals 2. It means that we have only two phages for one bacteria cell. We see that the differences between blue and red. Blue is a control trial and red is a research trial, which is we have overproduction of these two molecules together because they are really close. So we made just a construction with these two together, and we see that yes, the bacterial growth is less, so it means that it's faster. The lytic cycle is getting faster, and what is important for phage, not only the faster time of going to the lytic cycle, but also the efficiency of disorganization, which we have considered that by taking samples from the 270 minutes incubation with MOE, a multiplicity of infection equals to just the samples from here this time, and count the colony forming units per milliliter. And what we get that survived cells is significantly differs. There are significant differences, what was clarified by this previous research, but also the lysogeny among survivors is also differs. Efficiency of disorganization in normal phage is about 55%, and with the overexpression of these molecules, efficiency of disorganization is less. Is quite less. It's 18. Efficiency of disorganization, maybe I should explain it before, is that parts of these bacteria by yellow squares is a part when we have a prophage. When you have these ones, we have prophage. So when you don't have a prophage, the lytic development was started. Depending on our results, we propose mechanism of action of APROI1. It may act to prevent premature termination in T1, thus allowing for transcription of ROI gene coding for antirepressor ROI gene coding for antirepressor protein. As a result, the lytic phage development may be probably stimulated by ROI-dependent inactivation of C1 repressor and leads to observed changes in bacterial culture growth and disorganization efficiency. What I said, that's the decreased bacterial culture growth and less effective disorganization. Conclusions. APROI1 and APROI2 may interact as both trans-acting and cis-acting siRNA molecules. Overexpression of APROI1 and APROI2 decreased the growth of Escherichia coli culture and also decreased the efficiency of bacterial disorganization with phage 524B, and these molecules are probably one of the first candidates of functional microRNA type molecules of phage origin. To be honest, there are only one which is confirmed confirmed existence. Okay, and okay, thank you for your attention.
Okay, thank you, Wojciech. Are there any questions to Wojciech? I can see. Okay, so I have one.
Okay.
I'm sorry, but I'm not a biologist. I have nothing to do with bacteria, but I'd like to ask you for what, where do you use these molecules and the research? Like I quickly Googled what does it mean, and it's some kind of alternative for antibiotics, right?
No. These molecules origin is 524B phage. Okay, we have found him in his genome, and the matter of that is that, you know, I go back that the Shiga toxin production takes only place in lytic cycle, and we need to find out how the lytic cycle starts in any way possible to get to find out how to manage with the enteromagic Escherichia coli. This is why we take care of this examined microRNA, because as I mentioned, antibiotics do not help, and only way to help, only treatment is only to put the patient to hospital and waiting that can manage it by himself. Is it possible to, when the case is really very hard, it is possible to give him an azithromycin, but it's really, it's the last chance for him, and this is last chance for him, because we know that the Shiga toxin level will be increased. So once again, we we researched these molecules to get to know how lytic development starts, to even predict when the Shiga toxin production takes place, and help somehow this way. This is not an application research. It's really basic for now.
Okay, thank you. Are there any more questions? Okay, then thank you very much, Wojciech. And this was the last presentation of our session. Now will be a lunch break, and we will meet here again at 12:45. Thank you for your attention, and see you soon.
Thank you.
Hi everyone, welcome back. We will start the next session, 3B. Our first speaker will be Alessandra Bicocca from the University of Milano, and she will tell us a little bit about the air quality assessment. Hi Alessandra, the floor is yours.
Thank you. Okay, I'm sharing my screen. Can you see my presentation?
Not yet. We can hear you well, but we cannot see the presentation. Okay, now it's starting, I guess. Yes, we can see it very well.
Okay, perfect. Thank you. Good afternoon, everyone. I'm Alessandra Bicocca, and I'm a PhD student in environmental sciences at the University of Milano Bicocca in Italy. And today I'm going to talk about one of the main fields of interest of mine, that is the study on the indoor air quality assessment for the cultural heritage. Okay, first of all, I need to introduce you the concept at the base of such kind of studies, that is the preventive conservation. In fact, I'm mainly a conservator and restorer of cultural heritage, and I well know that there is always an interaction between the cultural heritage and the environment where the cultural heritage is conserved. And it is of main importance to study the conservative status of the cultural heritage connected to the conservative environment. And we can do this with the monitoring of the main environmental parameters and the main pollutants. Environmental conditions of cultural heritage are among the most crucial aspects to be considered when we determine the location, we plan the assessing procedures or the preventive measures. And it is of main importance if we consider that we are living in a reality of environmental global change. And cultural heritage can be affected by, for example, atmospheric particles and gaseous pollutants, both indoor and outdoor. And this is of main importance for those works of art that are characterized by a precarious conservational state. And this is the reason why it is essential to monitor all environmental parameters, including the relative humidity, the temperature, and of course, the main pollutants. Now I can introduce the 3 case studies that we have investigated. The first one is maybe one of the most famous painting of all times, and you already know that this is the Last Supper. This is Last Supper, or Ultima Cena o Cenacolo in Italian, and this is a late 15th century mural painting by one of the greatest genius of all times, Leonardo da Vinci. And it is housed by the refectory of the convent of Santa Maria delle Grazie in Milan. And this is the original location, and this is one of the reasons why it had a sad conservative history, connected also to the strange painting technique that Leonardo used to make it. The second case study is the museum, Museo del Novecento in Milan, where you can find this iconic painting. It is an historical and artistic Italian icon made by Pelizza da Volpedo. It is an oil on canvas, a huge painting, and where Pelizza depicts a worker strike, and it is an icon because it represents the labor protests of the 20th century. The last case study is the Museum of Galleria Borghese in Rome, the capital city of Italy. It is a cultural heritage itself because it is an historical building, and it is literally full of works of art. And in the many rooms that you can find in this museum, you can observe this beautiful marble sculpture made by one of the genius of his time, Gian Lorenzo Bernini, where he represented the story of Apollo and Daphne that you can find in Ovid's Metamorphoses. These 3 works of art are conserved in 3 different Italian museums. The Last Supper and the Perfect State are conserved in 2 museums located in Milan. And on the contrary, the last sculpture that I show you is conserved in Galleria Borghese in Rome. They are quite different locations. In fact, on one hand, the Last Supper Museum is an historical building, but it is characterized by a high control level of the main environmental conditions and the main pollutants. It has an heat and ventilation air conditioning system and many different several filtering areas and a fixed instrumentation for non-stop monitoring of the main pollutants. In addition, it is characterized by strong restriction on the number of visitors, and you have to know that the visits last only 15 minutes. On the contrary, the Museo del Novecento, also in Milan, has no control of the environmental parameters as the Galleria Borghese in Rome, but the huge painting that I showed you before is conserved behind a huge glass display case, and it has a protective action against mainly anthropic degradation risks and for other pollutants that we are going to see in this presentation. The last case of Galleria Borghese is the worst environment that we have studied, because it has a passive control of the main environmental conditions, and it has no filtration system due to the fact that it is a historical building of the 17th century. In order to investigate these three museums, a specific experimental setting was employed, allowing to perform a continuous high time resolution monitoring activity. And in some cases, this was done with a total visual impact reduction solution that was a novelty in this field of the cultural heritage conservation, and it was introduced with the Last Supper campaign, where a pre-existing hole in the wall was employed to install a sampling plate connected to all the instruments that you can see in this figure. A quite similar solution was employed in the Museum of Galleria Borghese, where all instruments were collocated behind this fake I don't know, panel with a reproduction of a decoration. Here I summarized all the instruments that were employed during the three campaigns, with time resolution of 1 minute for the Last Supper Museum and Galleria Borghese, and 5 minutes for the Museum of the Perfect State. Well, what about the most interesting results? First of all, I reported here the mean daily trend of the PM10 concentrations in the three museums. And the first thing that you can see is that the concentration of the PM10 are higher in Galleria Borghese than the two other museums. But we can see other interesting information in these graphics. First of all, in the case of the Last Supper, in the Museum of the Last Supper, we can see the the effect of the filtration system during the nighttime with a total reduction of the PMX. But also we can see that during the daytime, during the opening hours of the museum, these concentration increase, both for the PM10 and PM2.5. And this is because visitors are the unique source of the particulate matter into this museum that is characterized by a filtration system. The same increasing trend could be seen in this graphic of Galleria Borghese, but the most interesting thing is can be seen in this graphic of Museo del Novecento. And where in the Museum of the First Estate, we saw that the trends of PM10 and PM2.5 were quite similar, quite the same. So we decided to put the instrument, the optical particle counter, outside the display case of the painting, and this is what we have seen. This is the effect of the display case, and this shows us that only the fraction under the PM2.5 can pass through the display case. So this is a filter for the painting. And another interesting thing, it can be seen here, where the concentration inside the display case and outside the display case are different, and the outside concentration is over the limit that the limit for PM2.5 into the museums. Again, what about the fractions of PM2.5 and PM1? These are the mean daily trends of these fractions. And we can see again that we can find the highest concentration in the Museum of Galleria Borghese in Rome, but we can also see that all the mean values of these fractions are below the value, the limit that is indicated by the Italian Istituto Centrale del Restauro and the American Society of Heating, Refrigerating and Air Conditioning Engineers. What about the black carbon that is part of the finest fraction of PM, and it has a light absorption feature? I reported here the trends, the mean daily trends of black carbon in the three museums and outside in the context of Milan as a comparison. And again, we can see a total reduction made by the air filtration system in the Museum of the Last Supper. And again, we can see that the two museums that hasn't a filtration system has the highest concentrations, and they are near to the outdoor trend that we can find in Milan.
Sorry, Alessandra, you have only 1 more minute.
Okay, I can't hear you very well. I'm sorry. Okay. Briefly, the last results. We also investigated the gaseous phases, the nitrogen oxides, and again, we can see that the total reduction of these phases into the Museum of the Last Supper and high concentration in the other two museums that follow the outdoor trends both in Milan and in Rome. The gaseous phase of ozone also follows the trends of the outdoor in the two cities, and for the two museums without a filtration system. And on the contrary, a total reduction can be seen in the Museum of the Last Supper. This is a comparison of the trends of NO2 and ozone indoor and outdoor in the two cities, where clearly during the day the concentration of ozone prevails, as the NO2 presented dissociation due to the photolysis.
I'm sorry, but it's time to sum up. Sorry.
Okay. I'm sorry. Thank you for your attention.
No, it's okay. It's a very nice presentation, but actually, you know, we have to stick
I'm really
to the
sorry.
plan. It's okay. There will be just later time for the questions, and we have to go further. I'm really sorry. Our next presenter is Anna Buch from the University of Gdansk, and she will say something about the precipitation in the coastal zone in the Gulf of Gdansk last year. Anna, are you with us? Anna?
Hello, one moment, I have technical problems.
Okay, so maybe we can change. Do you need some more time?
Can you repeat?
Maybe you need some more time. Is Damian Mindykowski there?
Yes, I think I need a little more time for my computer.
Okay, Damian, are you there? Can you?
Yes, I am here. Do you hear me well?
Yes, we hear you well. Would you like to present now?
Of course, I can.
Okay, so now Damian Mindykowski will talk us about the elastic waves and testing the structures.
Okay, do you see the presentation?
Yes, we can see you and hear you very well.
Okay, so my name is Damian Mindykowski, and I'm a PhD student in Polish Academy of Science at the Institute of Fluid and Flow Machinery. My supervisor is Tomasz Wandowski, because I'm involved in the project correlated with PhD studies. And today I would like to tell a few words about non-destructive testing of structures by means of elastic waves. So at the beginning, I would like to tell a few words about the outline of my presentation. So I'm planning firstly to tell a few words about some key information referred to the topic. Next, I would like to say something about different non-destructive testing approaches, the one which I'm involved in and the other ones. Moreover, I would like to focus on ultrasonics later, because this is the method I'm using at work. Next, I will tell a few words about what I'm doing every day, and some example works and results in terms of animations. That's what I want to present at the end minutes. So, key information referred to the topic. Non-destructive testing is a kind of evaluation process of a structure. It's just a type of maintaining the structure in order to avoid catastrophes and to ensure that the structure has its properties reserved as they should be. Main aims of non-destructive testing are just what I told, so the avoidance of catastrophes and moreover estimation of material properties. This is the other purpose of NDT. Detection of surface and inner defects, just to avoid these catastrophes. And beside of non-destructive testing in the past, it was called as destructive testing, but due to its limited possibilities in case of usage, due to obvious reasons, because each evaluated structure needs to be distracted for the testing, so we don't want it. And that's why it evolved into non-destructive testing later. Main applications of NDT are aerospace, building sector, and shipyard industry. In case of the programs correlated with non-destructive testing, there are different softwares used, and each software depends on the application and, of course, what license you have. Most common programs we use in this branch in our institute are COMSOL, ANSYS, and MATLAB. We use COMSOL and ANSYS for modeling of the phenomena, and MATLAB for signal processing of possessed measurement results. In case of the NDT absence, the consequences may be obvious. So most spectacular consequences are unfortunately catastrophes, and some of them are depicted on the current slide. So first relevant catastrophes are related with the aeroplanes, like it can be noticed now in the upper part of the slide. So the damaged turbine engine of the plane. Other catastrophes caused by improper or a lack of NDT are the broken wing of the wind turbine, or the fatigue crack in the rupture pipe, or even the bridge catastrophe. In case of the historical aspect of NDT, it can be said that at the beginnings of non-destructive testing are about 80-90 years ago, and the X-ray usage was the first industrial NDT application, although X-rays were developed in the medicine reasons, in case of the medicine reasons, but later it was also implemented in the industry for NDT. And at the graph placed in the current slide, one can notice the very rapid exponential growth of interest of different NDT methods within the last 30 years. So we can see that this is the branch currently being really well developing. Now I would like to tell a few words about common NDT approaches, as I mentioned at the beginning. So the first one is thermography. This is a common approach referred to infrared radiation, and the usage of this method requires to have the heat emitter and the heat detector. So to heat up the structure, and later a detector to check how this infrared radiation, I can say, propagates within the structure. And the anomalies in this propagation of this infrared radiation may indicate on the presence of different defects and other factors which are correlated with the structural changes. Another approach is ultrasonics. So this is the approach I'm involved in. It is mostly related to elastic waves propagation. These are a form of these are a form of waves propagating in the solid structures. These are elastic waves are physical waves, and just similar as in case of thermography, we analyze the propagation of these waves. We look for the anomalies in its propagation, and then we analyze it, and we try to investigate what defects are occurred inside of the structure. Sometimes the defects are even visible using naked eye. So the working principle of this ultrasonics relied on the generation and later detection of these elastic waves, and we have different devices for that, both for the waves generation and detection, what I will say later. Another approach, also commonly used, is eddy current method. So this method relies on the electromagnetic phenomenon, but it's similar to ultrasonics in this context that it relies on the generation and detection of ultrasonic waves, but in a different manner than conventional ultrasonics. So in case of eddy current method, electric current flowing in electric coil located close to the evaluated structure causes an appearance of eddy currents on the structure surface. And later, these eddy currents propagate through the structure in form of elastic waves. And transmitted and received signals are compared. The important limitation of this method, unfortunately, is the fact that it can be applied only for conductive materials. Here are mentioned another NDT approach, less commonly used, although it depends on the application, of course. And so this is terahertz method, radiography, shearography, acoustic emission, penetrant-based methods, and visual inspection. So the last two methods are the simplified approaches. In case of ultrasonics, on which I would like to focus now, first I would like to present some used equipment for elastic waves generation and elastic waves detection, which we use during our work. So the most important equipment for the elastic waves generation are piezoelectric transducers and air-coupled transducers. Sometimes pulsed laser can be also used. And in case of the equipment used for the waves detection, one can distinguish laser vibrometer and optical microphone, although we use laser vibrometer significantly more often than optical microphone. But this is just different approach, and both devices have advantages and disadvantages. The materials of structures assessed by elastic waves-based methods can be different. The most common materials assessed by elastic waves are aluminium, steel, composites, polymers, and even concrete in case of the building industry. And in case of the classification of the elastic waves-based methods, two main groups can be distinguished, so contact methods and non-contact methods. The word contact or non-contact depends and means that the generation, detection, or both generation and detection of elastic waves is performed in contact way, so keeping the contact with the structure. So you need to bond the transducer, bond the sensor, or couple somehow the sensor with the structure to make the method to be contact method. And if you have a non-contact method, then generation and detection of the waves is normally contactless with the structure, what has some advantages. So in case of contact methods, you can see ultrasonic, C-scan, it's a conventional method, point-by-point measurements through thickness of the structure. Besides photoacoustic method, piezoelectric transducers-based methods, and acoustic emission. In case of the non-contact methods, as I mentioned, both generation and detection of elastic waves need to be done without contact of sensors with assessed structure. We normally use air-coupled transducers for that, with a certain excitation frequency. So here are a few examples of elastic waves-based methods in practice. So our work is currently now mostly correlated with the example in the right side of the current slide, in the right upper side. So we have the plate, we have air-coupled transducers. In this example, one is generating elastic waves, one is detecting the waves. we normally use air-coupled transducer and laser vibrometer for detection, but it's something like this. So my everyday work scope. Main target of the research is analysis of phenomena related to air-coupled acoustic waves transfer into elastic waves. So we generate waves in the air in form of acoustic waves, and later, due to interactions of these waves with evaluated structure, they are converted into elastic waves. So I have experimental part and numerical part of the research. Here are the most common examples of the structures we evaluate in form of plate-like structures. So CFRP, aluminum material, and glass fiber reinforced plate, most commonly used in aerospace industry. So this is the presentation of my everyday workshop, I can say, or laboratory. Air-coupled transducer and scanning laser vibrometer, as I mentioned previously. And here is the model developed by my colleague in our research team to model the phenomenon of this elastic waves propagation in plate and conversion from acoustic waves into elastic waves. And at the end of my presentation, I would like to present some results, some animations from my measurements. Maybe I skip one because the time is running. Maybe I will show the PZT-based impact defects detection in the CFRP plate. So we bonded the transducer in the middle of the plate, and now you can see elastic waves propagating in this plate. So the plate is in form of rectangular shape, and within a few seconds you will see the defect. You see now southern lower part of the plate and upper part of the plate, two small circular type defects revealed due to elastic waves propagation. So it's something like this. They were generated in the middle of the plate, and in the upper side and within the lower side of the plate, the defects are revealed now. And we can estimate their shape, their size, and location. And last slide, another movie. This is ACT, air-coupled transducer, so non-contact inspection, square plate, and you see the mass type defect revealed within elastic waves propagation. So the previous presentation referred to contact method, and this presentation, this movie, I mean, referred to non-contact method of elastic waves generation. So elastic waves propagate, interferes, yeah. On the right side, you can see the RMS map revealing the scratch type defect. Some of the elastic waves are reflecting from this defect, telling us that there is a scratch there. Okay, so this is the list of references, and thank you very much. I hope you enjoyed the presentation, and if you have any questions referred to my research or to my work, you want to know something about the topic, then you can contact me by email or call me using the phone number on the right lower side of the slide.
Thank you very much, Damian. Thank you. It was really interesting topic. Unfortunately, we don't have much time for the questions, but you can always use chat. Yes, I recommend you this way. Joanna, are you here with us? Are you ready?
Yes, I'm here, and I think now it should be okay.
Okay, try to share the screen with us, and so the next presenter will be Joanna Buch from the University of Gdansk, with her presentation about precipitation.
Can you see it?
No, not yet.
Not yet. So
yes, I think it's almost. Yes, we can see it, not in the presenter mode, but you can just, yes, start the slideshow and we should see it. Just start the slideshow.
Is it now or not yet?
Not yet, but we may have some time lag.
And now?
Yes, we can see it very well and hear you. You can start.
Okay, so hello everyone. My name is Joanna Buch, and I want to show you some research results from a physico-chemical characteristic of precipitation in the coastal zone of the Gulf of Gdansk at the turn of 2019 and 2020. And maybe for the start, we have two types of depositions. It could be dry deposition and wet deposition. Dry deposition means that the air dusts and aerosols are settling down on the earth, and wet deposition is when the aerosols, dust, and ions are washing with the precipitations. And to classify the quality of the precipitation, we can use the Janssen scale. And in this scale, we have pH and EH. The pH is a quantitative scale of alkalinity and acidity of the solutions, and it's based on the concentration of hydrogen ions and in a given liquid. Conductivity, EH scale is a class of conductivity, and with the higher concentration of acidic pollutants, the conductivity rises. And the same is with, maybe not the same, but also with the pH class, the higher pH means that there are more alkaline ions in the solution, and the lower pH means that the solution is acid, and natural pH of rainfall is more or less 5.5 pH, and it's because of the CO2 we have in the air. And the EH of uncontaminated precipitations is from 0 to 15 microsiemens per centimeter. Factors influencing to the quality of the precipitation, we can say there are acid forming and alkaline forming. In acid forming, we have sulfur oxidized and nitrogen oxidized, and in the alkaline forming, we have ammonium ions, sea salt ingredients, and mineral dusts. The aim of the study was to determine the degree of contamination of precipitation collected in Gdynia at the turn of 2019 and 2020. The measuring station was on the roof of the Institute of Oceanography, which is in the Gdynia, there were two sessions, there were 2 periods where the samples were collected. It was a heating period between 27th of October 2019 to 1st of March 2020. And the second season was a non-heating period between 20th of July to 9th of September 2020. The precipitation were collected by collector type bulk. Then there were measurement of volume, pH, and EH of the precipitations. The next step was filtration with QMA Wetman filters, and the filtration was storage in the freezing for another analysis. The whole period precipitation volume was 1.8 decimeter cubic decimeter, and as we can see on the right, on the picture on the right, the heating season was the lower volume of precipitation than non-heating season, and it was the whole period and the heating non-heating period, there are average values of the precipitations. And the results was quite interesting because the heating season was higher values of pH and the lowest values of pH was in the non-heating season, and in the natural environment in our climate, it should be it shouldn't be in this way, because in the heating period we have more pollutants from the municipal sector, and this should and here we can see that it wasn't so that the heating season wasn't so didn't have so many pollutants as acid pollutants that non-heating season, and the conductivity was higher also in the non-heating season, and it is connected with lower pH. And the 46% of precipitation was the normal pH, and more than half was slightly elevated in the heating season, and in the non-heating season more than 30% was of the precipitation has pH strongly lowered in the non-heating season, and only 70% of the precipitation was with normal pH. And conductivity, nearly 60% were very high conductivity with many pollutants acidic forming. And here we can see the the pH and EH results for the I'm sorry, for all the periods and where the rainfall was collected. And we can see in the red on the red part the heating season, and with blue part non-heating season. And we can see that the lowest values of pH was on the last part of the measuring period. It was the first days of September, and we can see that in this time we have also the highest values of conductivity. And why this is this way? It's probably because the 2020 was the year with COVID, and in the heating season there were many laws that we had to stay at homes. So it could many firms also didn't work. So the values of pollutants in the air were lower than in the previous years. And in the non-heating season, we can go to the, for example, to the holidays. And in the free city, we have many tourists. So the high values of conductivity and the lowest values of pH were probably connected with the transportation and the tourism in the vacation season. And the lowest values of the pH and the highest of the conductivity in the first days of September can show that this is the last time where people could went back home from holidays, and the school kids goes to the schools. So there were big the big part in this shape of pollutants could have the transportation sector. And the average value of pH in the whole period was 6.02. In heating season it was 6.12, and in non-heating 5.62. And the conductivity average values in whole period was 54.4. In the heating season 38.6, and in the non-heating season 73.9. So we can see that the conductivity and the amount of pollutants acidic forming in the non-heating season in the average was really very, very high.
You have 1 more minute, Joanna.
Yes, and in conclusions, in the 2019 and 2020 research period, precipitation over Gdynia was more polluted in the non-heating season than in the heating season. And this is evidenced by the almost 3 times lower share of normal rainfall and as much as 31% higher share of rainfall contained with acidic compounds compared to the heating season. In the non-heating period, the share of precipitation with a very strongly increased conductivity was found to be almost twice as high. And the increased traffic flow related to the holiday season and to the return of children to school was probably responsible for the degree of pollution in precipitation. And thank you.
Thank you very much, Joanna. It was a very interesting presentation. Unfortunately, we have no time for questions. You can always use chat. And our next speaker will be Sylwia Różańska, also from the University of Gdansk, with a very up-to-date topic, the effects of the pandemic. Sylwia, are you with us? Is there
Yes, I'm
yes.
here.
Okay.
Yes, just 1 second.
Okay, it's important that you're here.
Yes, I am. All right. Can you see my...
Yes, we can see your presentation. Not in the slideshow yet, but we can see.
Yes, is it visible now?
Yes, it's visible. We can hear you. You can start.
Okay, so dear audience, and did you know that the long-term prospects continue to predict that the world will further urbanize over the next decade, from 56% today to more than 60% by 2030? Every region is expected to become more and more urbanized in the next 10 years. My name is Sylwia Różańska, and I have a pleasure to present something about my research, which is, as you can see, contemporary transformation of post-pandemic city center, city of Gdynia, as a case study. In the very beginning, I would like to underline that I started to make a research on Gdynia city centers around 5 years ago. I wanted to define where exactly this city center is, because there is no obvious square or something. It has rather a linear shape. And I really wanted to find out connections between people and the city center. And then in 2020, a pandemic became, and everything what I did started to became to be simply not up to date. So I was really upset. But then I realized that no, that is a very good occasion to find out how exact connections between people and city center look like. So every time I present, it's important for me to underline that the city for me as a researcher is not just a research area or phenomenon. I mainly understand city as a process, something what is constantly happening as internal part of environment. Obviously, man has not developed a better organization of life than the city so far, and is connected simply with human nature. At every moment, the city becomes an unknown sum of new experiences. And cities are a reflection of people's minds, and city centers are the core of these reflections, in my opinion. And now you can see how Gdynia has changed during 100 years, from 1920 till 2020. Going strictly to the topic of the speech, I would like to say a few words about the Gdynia. I hope everybody knows where Gdynia is, but if not, it's a city in northern Poland and the seaport on the Baltic Sea coast, with a population around 246,000 people, and it's 12th largest city in the Poland, the second largest in the Pomeranian voivodeship, after Gdansk. So Gdynia, as I said, is a very young city with short but turbulent history. In just a few years, from a minor fishing village, it became an important economic center and a symbol of the reborn Poland. It was built very quickly in the middle of nowhere, and in the very beginning, planners had problems with complement the urban vision of the city development because of port, as I said. Then in Second World War became stagnation. And later, between 1945 and 1988, we noticed an abandoning of the traditional approach to planning center city areas. In 1990 and 2020, then we can see a new approach to strategic city planning, going for revitalization and progress of urbanization processes. And now the pandemic situation makes it possible to attempt a deeper understanding of the meaning of central space. Now very shortly about the city center itself, about meaning. A city center is generally understood as a specific point, line, or area with the highest concentration of services and social activities. The center is characterized by the greatest concentration of super local services, what is very important, and as well as the culmination of super local processes. The main layers of the modern city you can find material and non-material are administration, services, transport and mobility, culture, branding, society, and identity. So what's the impact of the pandemic on the reality of Gdynia city center? For sure, the service function was limited to necessary services. About half of the inventoried premises of Świętojańska Street, which is one of the main streets, were closed. Social activity was reduced to a minimum. Cultural institution had been shut down. The administrative function has shifted largely to a remote mode, and I mean the hardest lockdown right now. So what I did a survey conducted between at the end of 2020 via internet. The survey involved 88 residents of Gdynia, and finally 79 questionnaires were valid. My main goals were to determine the area that respondents considered to be central. The following, what I asked, for example, which districts in your opinion are in the center of Gdynia? Which objects, buildings located in the space of the center of Gdynia come to your mind first? Give the names of the streets located in the space of the center of Gdynia, which are in your opinion the most important, and so on. Another goal, to find out the emotional connections between the residents and the areas they define as a central. And in order to this, the respondents answered questions such as, do you think that Gdynia city center plays an important role in the structure of the whole city? For what purpose do you use the city center? How important is the time you spend in the Gdynia city centers for you? What impact does this time spent in the Gdynia city center have on you? And the third key objective was very important for me, to investigate the actual strength of connections between Gdynia's inhabitants and the city center, and to identify potential problems occurring there. So what I asked is how COVID-19 pandemic lockdown of March-April 2020 and associated restriction on use of Gdynia city center affected on you. So what is very important, that the space of the center of Gdynia is for the respondents mainly an area for shopping and dining. Social meetings, also mentioned relatively often, belong to activities of lower rank, and they are connected with previous two. Sport and recreation are mentioned only occasionally. Is everything okay? You can hear me?
Yes, we can hear you.
Okay,
it's okay.
Sorry. Okay, so 70 respondents said that in their opinion, Gdynia city center plays an important role in the structure of the whole city. How important to you is the time you spend in Gdynia city center? And now 25 said it's important, 31 is very important. How do you feel about being in the city center? How it influenced on you? So for 31 people, it has positive influence, and for 23, very positive. And now you have comparison. The obtained answers raise the question about the quality of the relationship resident space. Why? Because almost half of the respondents declaring that the time they spend in the center of Gdynia is important or very important to them did not feel a decrease in their quality of life as a result of the radical restriction introduction on lockdown time. And again and again, more than a half people declare that the time they spend in the city center of their hometown has a positive or very positive influence on them, did not feel, again, a decrease in the quality or comfort of life as a result of losing the possibility to use those areas. So in total, more than a half people of surveyed do not declare a decrease in the comfort of life as a result of losing the possibility to use space, which is potentially an important element of their daily functioning. How I investigated, I simply asked, how do you feel about it, about this restrictions? So the question arises whether motivation to spending time in the space of Gdynia city center is strong enough. Is it rooted in the need to build a sense of belonging and local identity? At this point, it's worth returning the question of the purpose for which the respondents go to the city center. I already mentioned that the most important for them were services or shopping, sometimes social meetings and recreation, but this indicates that most of these needs could be satisfied in the close area of their places of residence or via internet when it comes about shopping. So for many respondents, the city center did not turn out to be irreplaceable space. None of respondents indicated the city center attracts them for purely aesthetic reasons. This issue is undoubtedly a field for further, maybe deeper scientific exploration. So in this moment, it's worth to mention the study created by Eukaryotic studio, which showed that each city needs certain universal recommendation, and their implementation must be supported by individual analyses, especially in extreme situation, which is pandemic. And that's why my research now for me is very important. So therefore, we can say that the leading idea in creating the center of Gdynia should be not treating this space as a neat matrix of simple human activities, but a multicolored board with a range of possibilities for gaining experience and cultural exploration. The observation of changes that occur in the use of public space in the city center should be a source of information on how its further development should be created. So now a short few words about conclusions. Then, as I said, more than a half of respondents describing the space of Gdynia city center defined by themselves as important in their everyday life do not feel a decrease in quality, comfort of life due to the limited access to the central space. So I think that sharing knowledge and experiences is a key to strengthening a sense of identity and thus creating a high quality central space that people will long for. The Gdynia city center should be created as an area of exchange of ideas and information, and only in some way as a service area. As an urban form, it should be created as a specific structure connected to specific types of exchange and meeting that will stimulate real development of this space. So thank you for your attention and feel free to ask questions.
Thank you very much, Sylvia, and thank you for keeping the time regime. Unfortunately, our session is we're already late, but all the presentations were so interesting that it was really worth to hear it. If you have any questions, please use the chat and we will see each other at 2 o'clock. Thank you again.
Okay, it's 2 o'clock. Hello, everyone. I would like to, as a session chair, my name is Dorota Majewicz. I would like to introduce a presentation entitled Spawning Zones in Atlantic Cod Otoliths, Underutilized Life History and Maturity Recorders by Dhanushka Institute of Marine Research, Norway. Do we have you here?
Yes. Can you hear me well?
Yes, I can hear you well, so I'm going to stop my camera and mute myself. Okay, so the floor is yours.
Yes. All right, is my screen showing up nicely or all good?
Yes, we can
yes.
see. Yes, it's fine.
Is it, can you see my screen or?
Yes, it's fine.
Yes, we can see.
Yeah, okay. All right. Hi, everyone. My name is Dhanushka. I'm a researcher from the Institute of Research. Oh, my internet is okay. Yeah, I'm a researcher from the Institute of Marine Research. And today I will be presenting some of the research from my PhD that I very recently defended, entitled Spawning Zones in Cod Otolith, Underutilized Life History Maturity Proxies. So just very briefly to introduce I think it's lagging a bit. Okay, no. Okay, just very brief introduction on fish metabolism. So metabolism is maintenance, growth, and maturation. And metabolism is influenced by multiple factors. First and foremost, the genetics, life history traits. So whether fish is maturing early, if it's reproducing early, if it's migratory or not. And in a second time, the environment. So typically whether it has high resources, if temperature is cold or high, if population is big or small. So all these factors will have effects on how an individual grows and matures. And that's the reason why some populations of the same species might have very different life histories because they live in different. And one very important tool in marine fisheries is the otolith, because the otolith is what we commonly call a biological recorder. So what are the otolith exactly? These are small stones found in the head of the fish. They are calcium carbonate structures, which are commonly used for sensory and orientation. But in marine research, they are extremely used, partly because of their ability to biologically record everything happening in the fish. So for a given individual, they will record changes in metabolism. In addition, they are also metabolically inert, which means that even after they've been extracted from the fish, they could be stored for dozens or even hundreds of years without even deteriorating, meaning the information, the biological information they hold on a given fish is available at any time. And it's even used in paleoscience to look at fish extinct for millions of years. So I'm not going to go into details of the amazing things we can do with otolith, but this ranges from looking at individual age, using morphology to discriminate between population, using isotopes to look at environment, or using trace elements to look at migratory pattern. What we will focus on here is the structures, more precisely, specific structures found inside the otolith once you cut it in the middle and look at the rings. So the structures in questions are what I call the spawning zones. And these spawning zones have been around for a long time, but they have not been extremely used outside of Norway. They were first coined by a Norwegian scientist called Gunnar Olefsen in the 1930s, who, when he was looking at otolith from very old cod from the Barents Sea, realized that most of them had a kind of pattern in the older rings, where suddenly the rings were getting very narrow, regular, and with absent from young fish. And because we know that the is okay, yeah. Because these zones were absent from the young fish, and because the cod from the Barents Sea is migratory, meaning that it traverses thousands of kilometers to spawn, therefore different environment, metabolic cost. Rolfsen say that perhaps these so-called spawning zones were, these narrow zones were spawning zones. So they were representative of the years where a given individual would migrate and spawn. But since then, although they've been recorded a lot in Norway and used a lot, they haven't really been studied or even talked about in any other place in the world. So just to illustrate what these spawning zones look like, here you have two otolith from 10 years old fish from the Barents Sea. And the top one shows no spawning zone. You see rings are getting small each age because the fish grows less and less, but there's no specific pattern to be seen. On the other side, for the bottom image, we can see very well that the last increments are suddenly getting very regular, very narrow, with all significantly thicker translucent zones. So the translucent zones are the darker zones. This is kind of the typical spawning zone setup as currently identified in Norway. But as I said, they're not really looked at or even studied in any other place in the world. So around the world, when we look at maturity, we don't look at spawning zones, actually. We look at commonly macroscopic visual examination of the gonads. So this is very straightforward. Open up the fish, look at the gonads, stage them based on various criteria. And then from that, you're able to say a given individual fish is spawning, is juvenile, is resting. This is very effective because it gives a lot of information, but it has two major flaws. One is that the data is not saved, meaning you throw away the gonads afterwards, so you cannot really know and go back to the gonads and look, was it actually spawning, or maybe I looked at it wrong. And on the other hand, if you look at gonads from very old fish, it will only give you in the year, perhaps a year before maximum. So a 10-year-old fish, you will look at its gonad, you will know what the status is in the given year, and you will know what it was perhaps the year prior, but you won't tell if that fish was mature 5 years ago, which is spawning zones are a real thing. You should be able to do so. So all of that makes it very interesting to look at these spawning zones and see if there are a local weird thing happening in Norway, or if they might have actual potential. So for this study, we divided three main objectives. First one was evaluating whether the spawning zones could be identified in other cod populations. Secondly, do they actually form in relation to individual maturity and spawning, as it has been suspected for the last 10 years? And finally, if they do seem to form in relation to maturity, could we utilize them to provide reliable estimates of maturity? So to that extent, I collected about 1,200 totally from 6 different cod populations. The two first populations being the kind of population in Norway, where we know spawning zones are happening, but then also cod from the North Sea, from the Greenland, and also for Cape Breton, which is on the other side of the Atlantic. And in all of these cases, we also sampled multiple age classes that covered both the immature and the mature stages. So we knew for sure we had otolith from fish that were to be mature and fish that were old enough to normally be mature. To prepare to the discussion, point one, can spawning be identified in other cod population? Overwhelmingly, the short answer was yes. We could reliably identify spawning zones similar to those found in the Barents Sea population in all of the cod populations we investigated. This is even more interesting in the sense that these spawning zones be reliably identified by different age readers. Done in complete blind condition, all of the images were provided with zero information. So the readers did not know anything about the population, the cod image, or anything else. That proves that this definitely are a specific feature for Atlantic cod and not only of one given population as it was suspected in the past. But do they actually form in relation to individual maturity? Well, to test that, look at the x-axis. You have the proportion of mature fish on a given age class based on this derived from ads. On the y-axis, you have the same proportion derived from spawning zone. So what I've done is splitting all these respective increments. If it was not a spawning zone, then fish wasn't mature at that age. If it was a spawning zone, then the fish was mature at that age. And we aggregated that for all of the population per age class. And what we notice very easily is this specific U-shaped relationship where for the young and old age classes, the spawning zones reflect really well the maturity as expected from gonads, meaning that for the young fish where 0% of the population is supposed to be mature, well, none of them have spawning zones. And for the old fish where nearly 100% of the population is supposed to be mature, well, all of them have spawning zones. But in the intermediate age classes in the middle, where only a small proportion of the population is supposed to be mature, we see an underestimation from the spawning zones, meaning that less fish have spawning zones than those that are mature based on gonads. The main hypothesis we have so far is a biological offset. because in Atlantic cod, we know that the time of maturity is usually very poor. There's a very low reproductive investment. And alternatively, usually the second year following maturity is matched with very high proportion of skip spawning in Atlantic cod. So all of that makes us suspect that perhaps what's happening is that for the creation of spawning zones, it requires very strong metabolic changes, which are not taking place yet at maturity, because the first maturity is usually a bit so-so, and therefore there's not enough metabolic changes to induce the formation of these spawning. But a lot of intermediate classes, what we see here is that there is a very good match between these spawning zone formation and maturity from gonads. Therefore, there seems to be a definite link between spawning zone and maturation. So that leads us to the third objective, which was, if we know that they can be formed in relation to maturity, could we actually use them to give reliable estimates of maturity time series? So to answer this third and final question, what I did is I went back to Norway, where the Institute of Marine Research has a century-long autoliv database. And because in Norway, spawning zones have been known for a century, they've been recording the different spawning zone calculation for all of the different individuals they have. Therefore, I was able to extract spawning zone information from nearly 400,000 cod autoliv that were collected between the 1930s and the recent period in 2015. And from these spawning zones, I used the same methodological approach to split them into respective years and increments, and then recalculate the proportion of mature fish for a given age class for each year. So what you see here on the figure to the right is so the proportions for each age class for each year. And in blue, this is what we estimated using spawning zones. And in black, this is the reference using the ogives from the stock assessment. And what we see very easily here, it's even more clear than the previous figure, is that the reconstructed time series of maturity using spawning zones are extremely close to those using gonads and show identical viability through time, which is a definite proof that these spawning zones are reflecting some things happening with the maturity of the individuals. What we do notice, however, is also this specific U-shape I mentioned earlier, which is here confirmed again with underestimated proportions of mature fish at a given age, especially for the intermediate age classes. If you see the two black circles on the figure, for age 7, which is typically the first year of maturity for Barents Sea cod, we see that the spawning zones underestimate quite a bit the proportion of fish that should be mature at age 7 for the given years. While for age 10, which is a year class, an age class, sorry, where usually almost all of the population is mature, we see that the curves are extremely close. So what can we conclude from these three results? First of all, the spawning zones are universal to Atlantic cod, irrespective of life history, migratory behavior, and environmental conditions, which were some of the traits that were suspected to induce formation of spawning zones prior. So now in the future, what we would like to look at is whether these spawning zones can be found in different species or even different families. We already have clues that they might be common to all gadoids, mainly because they are used in Norway and found in hake and haddock. So I would like in the future to see if we can find them in different species. They're also tightly related to the onset of maturity for sure. They might not be a one-to-one proxy of actual spawning event, but they definitely start forming after the onset of maturity. And we have a visible offset of 1 to 2 years following the first maturity. And knowing the biology of cod with poor reproductive investment in the first time spawning and frequent skip spawning in the second mature year, our lead hypothesis so far is that the spawning zones might be formed in association with a so-called delay acclimated energy allocation following maturity. What that means is that once the fish gets to maturity, his metabolism starts changing. But because the reproductive investment for the first 2 years is a bit bad, it actually takes some time before the metabolism really changes and then starts consistently inducing spawning zones in the autoliv. And finally, they are a very effective proxy for maturity time series, and that could have very high potential when autoliv archives are available. Typically for data pool species where we have very little information, but we might have autoliv archives available. These could be used as secondary proxies. And alternatively, when we have long time series of autoliv, but only shorter time series of maturity, we might be able to use these autoliv archives to extend in the past the historical variations in maturity that we might want to investigate. So yeah, altogether, this should be underlies potential of spawning zones, which before this study had been virtually never really studied and never really talked about. So I hope there's some really interesting things to look at in the future regarding these spawning zones, and not only in cod, but in any fish species. And that's it for me. Thank you for your attention. And if you have any question, please go ahead. I will be happy to answer.
Okay, thank you very much. Do we have any questions from the audience? Okay, thank you, Monica. Okay, if any questions appear, surely you can text Com and ask him any question later. Okay, thank you so much, and thank you for the perfect timing. It was a quarter past. Okay, could we? Thank you so much. Okay, thank you. Could we please ask Claudia Szabelka? Do we have Claudia here? Yes. Okay, Claudia is going to tell us about integrating a human rights-based approach into economic growth through the realization of the right to development, inevitable conflict, or a place for a common ground. Claudia is from the University of Glasgow. Claudia, if you're ready, the floor is yours.
Thank you very much. Can you see the slides, and can you hear me well?
Yes, I can hear you well, and we can see the slides, yes.
That's perfect. Thank you very much. Good afternoon, everyone. Thank you, everyone, for attending this session and showing your interest in the topic. My name is Claudia Szabelka, and I'm going to present to you my research results, integrating a human rights-based approach into economic growth through the realization of the right to development. Finally, we are going to answer the question of whether this nexus causes inevitable conflict or is a place for a common ground. I am currently reading a master's degree in social sciences at the University of Glasgow in politics and economics. I'm particularly interested in development research in those particular two areas, that is human rights and economic growth. To start with, this research topic is an increasing in importance problem, especially when considering the recovery post-COVID-19, increasing concerns of human rights, as well as it is an important aspect to consider for developing countries. And finally, crucial to answer the question where the world is heading. As both human rights and economic growth are increasing in their importance, they are considered in the most crucial goals for Sustainable Development Goals, that are SDGs. It is because both are recognized as key aspects of Sustainable Development Goals. During my research, I discovered that when considering human rights aspects, all of 17 goals relate to them in some instance. However, the visibility is not as formal, and so they do not have a dedicated separate goal. This will be explored later in this presentation. When considering economic growth, we do have a dedicated SDG growth that is very extensive with all targets formally set out. Let us focus now on human rights. What are they? Why countries need them? Human rights, in short, are norms and moral principles that are inevitable, inherent, and universal. The Universal Declaration of Human Rights, adopted by the UN, was the first legal document set out the fundamental human rights to be universally protected. Ensuring universal human rights is an important step we must take to recognize human rights as features of our everyday existence, is to make our lives worth living. And they are the basic ingredients of our good life. If we look at SDGs goals, goal 16, that is peace, justice, and strong institutions, we can see that solely this one goal refers to 6 human rights, that is right to access to information, right to participation in public affairs, and for example, right to legal personality. And it relates to over 20 article records in international human rights documents. And this is really an enormous number and is very similar when considering all the rest goals. Now let's look at the descriptive statistics of human rights indicators in the world. On the right, you can see the index of economic freedoms that considers labor freedom. And on the left, there is the map of international coordinating committees of national human rights institutions. And dark green means that there are Class A institutions. So if we look at two maps, the regions where there's no color or like or very light red color, it means that the human rights are worse. So you can see, for example, it's in the case of Africa and South Asia in those two maps. Let's please focus now on this slide for a moment. I would like you to focus on the areas in the circles. We will later going back to them, and they basically indicate pure human rights. If we look into economic growth, this is how it's measured and its importance. Economic growth is the geometric annual rate of growth in GDP among countries. It's targeted by SDG 8. It aims to promote inclusive and sustainable economic growth, employment, and decent work for all. Sustained and inclusive economic growth can drive progress and create jobs and improve standards of living. Now again, if we look at the map, we can see that regions that were previously indicated as having pure human rights have high economic growth. What will eventually sounds very bad and highlights that the proportion of growth in those countries is achieved through violations of human rights. Therefore, if we look at the map's descriptive statistics, there will be something very easy to notice, that is economic growth is not equal human rights. Of course, there is some caveat in the literature. Some studies explain that this relationship will be in particular true among developing nations in regions like Asia and Africa, whereas in the Western countries, human rights and economic growth will be positively correlated. However, it all heavily depends on what indicators of human rights were used. If they consider indicators like torture, violence, trafficking, corruption, then yes, that is true. However, if we consider aspects of human rights like right to safe drinking water, right to education, right to health, and free expression, then the positive correlation in Western countries can become more vague. So does the solution exist? Can we have both improvements in human rights and economic growth at the same time? 30 years ago, the international community came together to debate the challenges affecting our world. Until then, development had been equated with economic growth. Only in 1986, the UN adopted the Declaration to the Right to Development. Therefore, one way of assessing the relationship is through the realization of the right to development. Nowadays, references to it can be found in all major UN documents, including the Millennium Declaration. Right to development can be described as an integral part of fundamental human rights. However, its implementation has been very slow due to the highly politicized nature of the issue. This declaration defines development as an economic, social, cultural, and political process aimed at the constant improvement of well-being of the society. It made development the right of all individuals and peoples, called for active, free, and meaningful participation in development and fair distribution of its benefits. It rejected foreign domination, racism, discriminations, and all violations of human rights. Therefore, it seems that this solution is already out there, and the right of development addresses both economic growth and human rights. However, there are some problems with it. Although it's a very powerful idea, that can be applied, it has the legal status of soft law. Soft law in international law is a law that is generally accepted but is not legally binding. What does it mean? What would mean the full implementation of the right to development as well? Then the developed countries are very anxious about it, because the right of development could be seen as a right to everything, and allowing states or individuals to sue rich nations for fulfillment, for example. There is some other resistance to this idea as well. Some states tend to express rhetorical support for this right, but neglect its basic precepts in development practices. Like the United States opposes or is reluctant to recognize development as an international human right. And yet the Obama administration has proposed nearly double its development spending. However, it's reluctant to have right to development as a legally binding force. For example, Germany fully endorses the concept, however, of the right to development. That means growth with equity. However, it doesn't acknowledge the principle as a binding right as well. Another example can be African Union that regrets the absence of a clear right to development based approach. It agrees that national action and international cooperation must reinforce each other, but calls on donors to operationalize the monetary consensus. Therefore, the ongoing work within the frame of the United Nations is giving a rise to a common understanding of the right to development. One major example of the process is the constructive working spirit that dominates the debate, as well as the inclusive approach in which the debate is being conducted. The fact that the delegations from different regional groups, development organizations, and international financial institutions are engaged in a fruitful discussion gives hope for further positive development. However, the donor countries are hesitating to commit to a right whose content and interpretations are still under discussion. This again shows why the debate at the UN is so important. Some may have criticized it for being overly theoretical and lacking examples on the ground. However, first, the debate is advancing towards criteria for application. Second, this theoretical debate has to be conducted and needs to come to conclusions that are applicable for all parties involved, because this is the only way to convince reluctant parties to give a real chance to the application of the right to development on the ground. And it is only this application that can enable the right to development to serve the purpose it was formulated for. Therefore, change the lives of people and become a reality for everyone, freeing the entire human race from want. Thank you very much.
Thank you very much, Claudia, for your presentation. All right, do we have any questions from the audience? Claudia, may I have a question then? Since you mentioned that there is a problem with defining, a practical application for the definition of human rights, what do you think stops, for example, United Nations or any other worldwide organizations from defining it? Is it really practically so hard to provide a reliable, sensible, and definition that would be clear so that the human rights could be applied, right? Okay.
So with human rights, there are many documents that the UN created that are providing some definitions of what is human rights. So human rights are basically all are well defined. However, the problem with the UN is that most of the documents are soft law. So this means that the framework is already there. However, there's no legal formally binding force. So nations should follow the patterns and the legal framework that is there. However, if something is not working or they don't follow it fully, they don't have any sanctions, or like they are not binded by this legally fully, because it's kind of a soft law. So it's up to the country if they decide to follow it or not. However, the framework is already there, but yeah, because of the highly politicized issues, it's not formally binding, because, for example, some countries are donating money to authoritarian regimes where human rights are violated. So those countries basically don't want to be binded by those human rights laws, because then it would mean that they are donating basically to countries that violates human rights, and they are like in their policies saying that they are very good for people, So this is something that is in conflict for them. So they don't want it to be binding. So they would prefer to stay on a soft law basis. This is very sad, actually, but this is how it works in international law, sadly.
Actually, the fact that the law cannot be enforced is actually puts everything into the question. But I agree. Okay, thank you so much. Do we have any more questions? Okay, if anybody wishes to ask a question to Claudia, please, you can place it in the chat. So thank you, Claudia, for your very interesting presentation on a very contemporarily important topic. There has been a lot discussed on the human rights, and I hope a lot more is going to be told and done, said and done about this. Okay, thank you so much. Now I would like to go on to Liliana Sharma's presentations, pharmaceuticals in the aquatic ecosystems.
Let me know if you see the title of the presentation and you hear the voice when I... Did you hear the voice?
A little bit, not much.
Let me do it again, okay?
A third-year PhD student at the Institute of Oceanology, Polish Academy of Science.
Yes, we can hear it now.
Now it's better. Okay, so
yes,
I started it from the beginning and mute myself.
Me too.
Good afternoon. First of all, I'd like to introduce myself. My name is Liliana Sharma. I'm a third-year PhD student at the Institute of Oceanology, Polish Academy of Science. Today I'm going to give you a general overview of the problem of the presence of pharmaceutical residues in the aquatic environment. First, I would like to start with a short introduction. Micropollutants are substances that occur in surface waters in very low concentrations, and their impact on human health and the environment is not well understood. One example of micropollutants is pharmaceuticals. They represent a major group of emerging pollutants found in freshwater and coastal waters. Pharmaceuticals are essential for human health and well-being. However, the growing use of pharmaceuticals has become a new environmental concern due to their potential negative effects on humans and ecosystems. About 2,000 active pharmaceutical ingredients are being administered worldwide in prescription medicines, over-the-counter therapeutic drugs, and veterinary drugs. The vast majority of them have not been evaluated for their occurrence, fate, and possible impacts on water quality, human health, and freshwater ecosystems. Pharmaceuticals in the environment are a challenge to manage for the following reasons. Pharmaceuticals are designed to interact with a living system and produce a pharmacological response at low doses, which makes them of environmental concern even at low concentrations. They are designed to be stable and persistent, and for most wildlife, exposure to pharmaceuticals in the environment could be long-term, potentially occurring via multiple exposure routes and involving a mixture of substances. Managing the risk of pharmaceuticals in the environment requires a multidisciplinary and multi-stakeholder approach. The global pharmaceutical industry is estimated to see consistent growth due to the rising and aging global population, increasing income levels among poor and middle-class families, rising access to quality healthcare, and the emergence of new diseases. As per United Nations, the global population is expected to cross 9.3 billion by year 2050, and around 21% of this population is expected to be aged 60 and above, representing a large customer base for pharmaceutical products. To manage the risks of pharmaceuticals in the environment, it is first necessary to understand the origins, entry pathways, sinks, and concentration patterns of pharmaceuticals in the environment and their effects on human and ecosystem health. Pharmaceuticals are present in the environment as a consequence of pharmaceutical production and formulation, patient use, use in food production, and improper disposal. Figure 1 presents a schematic diagram of the known contamination pathways related to the use phase for both human and veterinary medicinal products. Research shows that between 30 and 90% of the orally administered dose of pharmaceuticals are generally excreted as active substances in the urine and feces. Removal rates for pharmaceuticals in wastewater treatment plants range from less than 10 to almost 100% and depend on the characteristics of the pharmaceutical and type of treatment technology. Conventional wastewater treatment plants are not designed to remove pharmaceuticals. Furthermore, veterinary pharmaceuticals used in agriculture and aquaculture can enter water bodies directly or via surface runoff. To sum up, the main sources of pharmaceutical residues in the environment are pharmaceutical manufacturing industry, consumers including excretion and inappropriate disposal of pharmaceuticals to wastewater systems, hospitals, and agriculture. Humans can be exposed through drinking water and ingestion of pharmaceutical residues in plant, crops, fish, dairy products, and meat. The continuous and increased production and use of pharmaceuticals have led to their widespread occurrence in the aquatic environment across the globe. Many pharmaceutical residues have been found worldwide in soils, sediments, surface water, groundwater, and drinking water. For example, a team of researchers tested 711 river sites in 72 countries for the presence of antibiotics and found antibiotics in 65% of them. On 2003 data, over 2.3 billion people live within coastal limits, representing 31% of world global population, and more than 50% of coastal countries have 80 to 100 of their total population within 100 kilometers of the coastline. Global demographic trends suggest increasing numbers of people living along coastlines. These trends suggest the potential for increasing inputs of human pharmaceuticals into coastal environments and therefore the need to address potential exposure scenarios and implications for marine risk assessments of drug residues. The Baltic Sea ecosystem is particularly sensitive to pharmaceutical pollution because of its low biodiversity and many species experiencing increased physiological stress due to the brackish water environment. The water exchange rate in the Baltic Sea is slow, meaning that there is a long retention time for persistent substances. This makes the Baltic Sea ecosystem more sensitive to hazardous substances in comparison with other marine areas. It is challenging to assess the potential long-term risks of trace amounts of pharmaceutical in the environment. The vast majority of pharmaceuticals have not been evaluated for their long-term toxicity, occurrence, or fate in the environment. Antibiotics are a specific group of pharmaceuticals of potential ecological and human health concern. Antibiotics work miracles. In the human body, antibiotics target infections and prevent diseases. However, once they are excreted from our bodies and dispersed into waterways, antibiotics can harm the environment. One way this occurs is by promoting increased microbial resistance in the environment. Antimicrobial resistance is a global health crisis with the potential for enormous health and economic consequences. Antimicrobial resistance occurs when microorganisms become resistant to antibiotics, antifungals, and antivirals. This phenomenon poses severe risks to global health, livelihoods, and food security. Drug-resistant infections are estimated to cause 700,000 deaths each year globally. Additionally, aquatic organisms are exposed to waterborne contaminants during their entire life cycles. Antibiotics may induce chronic effects such as changes in behavior, reproduction, and growth. Long-term alteration of bacterial community composition may lead to variations in biogeochemical cycling in aquatic ecosystems. For example, anoxic environments promote harmful algal blooms. Antibiotics may have bactericide and bacteriostatic effects, with the consequent disappearance of some microbial populations and their ecological functioning. Microbial biodiversity is important for maintaining biological processes in water and soil. Despite growing public concern over antibiotics in the natural environment, governments worldwide fail to adequately regulate household and industrial effluent containing antibiotics. Growing natural antibiotic resistance affects both humans and animals. One great concern is the emergence of new superbugs resistant to common antibiotics and therefore difficult to eradicate. It is important not only measure the environmental concentrations of pollutants, but also whether they are toxicologically significant or insignificant. That is, whether they have the potential to cause adverse effects to human or ecosystem health at concentrations observed in the environment. The large number and variety of pharmaceuticals available mean that it is unlikely that we will be able to monitor and test all the substances. So it is important to identify the pharmaceuticals of the most concern. A key data required for determining the likely impacts is information on the number of pharmaceuticals used in different regions through better understanding emissions and pathways. When considering toxicity risks, several factors can complicate assessment. In real life, substances are not isolated in the environment. Instead, they occur mixed and in combination with other contaminants. Even if a given substance is at concentration too low to be harmful, when mixed in water with other chemicals, research has shown that the combined effect can be harmful. In the environment, transformation and degradation reactions alter the mobility, persistence, and fate of the pharmaceutical residues. Metabolites and transformation products represent a large amount of the total pharmaceuticals load. This is of concern since these compounds are rarely evaluated for their toxicity, occurrence, or fate, and some evidence suggests that the ecotoxicity of some degradation products can have higher toxicity effects than that of their parent compounds. Additionally, ecosystems and humans may be continuously exposed via several pathways of low-dose mixtures that can have additive effects. And that's all, and thank you for your attention.
Okay, Paulina, thank you so much for presenting Liliana's presentation. I guess if we have any questions, we can email Liliana. Okay, so if I could now please ask Monika Lenge, benthic efflux of dissolved organic carbon and its bioavailability, what is returning from the Baltic Sea sediments, biogeochemical perspective. We're all ears.
Hello, can you hear me? Two basic questions, if you can hear me and if you can see my presentation.
Yes, we can hear you and we can see the presentation.
That's awesome. Due to the poor internet connection, I decided not to show my face via the camera. So I put my picture so you can imagine who's talking. It's not just the voice from the internet. So hello everyone. Have you ever wondered what happens with all the water and matter coming into the sea? What happens to them? So today I will talk about what I like to call the Baltic hiccup. To understand the whole subject, we have to take a step back and begin at the top. The Baltic Sea is a semi-enclosed brackish sea with a catchment area 4 times larger than the sea surface itself. The exchange of waters with the North Sea is hindered by shallow Danish straits. It is under high anthropogenic influence, and huge loads of organic matter and biogenic substances enter the sea, especially via riverine discharge. It further leads to high primary production, which we know especially from green thick layers of algae that don't allow us to swim during summertime. We all know about rising carbon dioxide concentration in the atmosphere, what leads to global warming. Carbon dioxide has significant influence on the carbon cycle. CO2 is pumped into water, especially by biological process known as photosynthesis. It's called biological pump. As dissolved carbon, it enters the trophic chain, is changed into particulate organic and inorganic carbon, and falls to the bottom sediments. In the sediments, organic matter, mainly carbon compounds, is remineralized by bacteria, so it simply decays into less complex structures. Scientists thought for the very long time that what enters the sediments, it stays there forever, but they couldn't be more wrong. It returns from the sediments to the water, and we call it a benthic efflux of remineralization products. Going further to the depths of the sea, we have here an example of DOC concentration in the sediments. In the bottom water, we can sometimes have oxygen, so oxic conditions, but in the sediments we have no oxygen, so anoxic conditions. Oxygen is a crucial factor if it comes to benthic efflux. Less oxygen, the benthic efflux is higher. Let's have a closer look what happens in the sediment. Fresh organic matter from the sea and river falls to the sediments and is eaten and transformed by bacteria there. In the sediments, pore water moves towards surface, and in the uppermost surface, there we have a mixture of fresh and transformed organic matter. Now we know the basics of processes occurring from the top to the bottom. Now let's talk more about DOC. We know where it comes from, but what exactly is it? It's a dominant part of organic carbon in marine ecosystem, and it's a heterogeneous mixture of different carbon compounds. And what is its role in the marine ecosystem? It's mainly the source of food and energy for bacteria, and is an essential part of the carbon cycle. Why do we care about this subject so much, and how we got our answers from, you may ask? We went on the cruise at Oceania. We collected sediments, pore waters, and bottom waters from different parts of the whole Baltic Sea. But today let's focus on one side, the Gdansk Deep. It is a very interesting place for scientific purposes. It is a depositional area, so it means that everything what entered the sea most likely will accumulate there. And so it has a lot of organic matter, mainly coming from the Vistula River. After collecting the samples, we measured the DOC concentration. More was in the sediments and less was in the water. So basing on the simplest law of diffusion, water was pouring from the sediments to water, and voila, we had a benthic efflux. So you can see the amounts of organic carbon diffusing from the sediment. Less in the oxic conditions and more in the anoxic conditions. We also calculated the amount of dissolved organic carbon coming from the all depositional areas of the Baltic Sea, and it is 159 kilotons per year, and that's a lot. We know how much carbon is coming back from the sediments now, but we wanted to know more about fate of DOC in the sea. The subject is not so widely studied, so I give you our results, which are one of the very first in the field of biogeochemistry. So DOC decays, but since it is a complex mixture of different compounds, it is divided into 3 categories: labile, semi-labile, and refractory. The difference is basically how fast they are decomposed by bacteria. So labile and semi-labile are bioavailable, so easily used by bacteria as a source of food and energy. We did our experiment and calculated that 55% of pure water DOC coming back from the sediment is bioavailable. That's huge news, actually, and it can be used in biogeochemical models and introduced into knowledge about carbon cycle. Summing up, here we have the comparison of DOC from different sources. Now you can see that even if the amount of carbon coming back from the sediments is much less than the one coming from the rivers, it is much more bioavailable, so can be potentially much easier source of food and thus be reintroduced to the ecosystem. So we have to remember that what enters the Baltic Sea cannot be forgotten. It comes back and it has to be included into biogeochemical models and into carbon cycle. Sediments are a source of dissolved organic carbon, even up to 159 kilotons per year, and 55% can be potentially reintroduced to the ecosystem. And the last but not least, what is the possible future? In my opinion, the higher the temperature, because we have the rising temperature, the more primary production, the more they will see in sediments. So it's called a vicious cycle, or as I said at the very beginning, a Baltic hiccup. And that would be all from me. Thank you very much for your attention. This work was done in the framework of my own NCN project. And I encourage you to write to me if any question appear later or now, and go and see our publication if you would like some detailed information. Thank you very much.
Thank you so much, Monika. Do we have any questions to Monika? Okay. We have something in the chat. Okay, so we have just, Paulina has included some links. So now we're going to enjoy 10 minutes of a break, and we're going, more because we have finished earlier due to the lack of questions. Therefore, we have a break until 3:10, and then please let's welcome to the closing session, and Paulina has included the links in the chat. So let's go back to the main session after the break at 3:10 p.m. and I'll see you there. Thank you so much for your attention. And if you have any question to our speakers, please email them later. And thank you, all of you, for your presentation. See you later.